Good morning, everybody. Thank you so much for participating in this Zoom meeting. Today, my topic is organic hydroponic production of lettuce. Before showing our results, I would like to share the USDA 2019 organic farming survey status. I think the result is very encouraging and I wanted to share this. So certified organic farms and lands since they started to do the survey, as you can see from this table, especially from the last 3 years, from 2016 to 2019, the farm number increased a lot, and then cropland increased even more, the percentage. So you see, in the last 3 years, cropland is 30%. And the top 10 organic crops by sales in 2019 survey, number one is apple, but lettuce and spinach are also on the top 10. So that's very encouraging. These are the crops we are working on in our project. So the top 10 states, number of certified organic farms, California is number one, and look at all the states. They are all northern states, and all the southern states have much lower number of certified organic farms. Probably this is due to climate issues, and the hotter the weather, usually you would need to deal with disease and pests more. So, and there are limited product for organic farming control this disease and pests. And I think this is one of the issues. And of course, limited other product like limited organic fertilizers. The top states in organic sales in 2019, Texas is number 5. So that's good news. But this, I believe, if you look at the detail, there are all poultries and, for example, organic beef, organic chicken, eggs, and milk, and those are the main sales from Texas. The market value of certified organic product sold in the last 3 years, and um, again, the crop is 38%. And major challenges in certified organic farms, number one is regulatory, and that's many organic farmers actually they struggled and they told us this, and also the paperwork to get to the certification and afterwards needed to be evaluated. So those are the things farmers are afraid, and also the fees are high, and the organic crops. The price sometimes not as high enough to justify to get to the certification. Those are the struggles, and the production problems. Those are the areas that we, as a scientist, researcher, and extension specialist, we can help on that side. So, future production plans for the certified organic farms. 44% says they're going to maintain the current levels, and the 29% says they wanted to increase. So these are good news and encouraging, and 20% says they don't know. So summary of organic farming status, and these are from the survey results and also from our interactions from stakeholders and also from the literatures. So we can see that organic farming is one of the fastest growing segments in the U.S. agriculture, and organic crops are growing rapidly, especially recently. And certified organic agriculture accounts for 70 million hectares globally, and with over half of that total in Australia. By the end of 2016, organic agriculture is practiced in 178 countries. So for sure, organic farming globally is a trend, is a global trend. However, even though the interest is increasing, the productivity is lower on average in organic compared to conventional farmings. Um, uh, across many crops, that's the average. Factors causing this new low yield in organic farmings, we think number one is fertility management. In organic farming, there's no standardized organic fertilizers. And the ingredients of organic fertilizer or organic ingredients in organic substrate and some organic fertilizers are also localized depending on where what available. And the slow release, and the nature of organic fertilizer is the slow release nature, which means they need to have some time to release, to mineralize, so that the fertilizers can be changed to the form that plants can absorb. And that release rate usually does not match the demand of crop needs, and especially in the conditions, for example, low temperatures, that would also affect the release rate. And limited organic products, including organic fertilizers, and for propagations like organic substrate, organic potting mix, and all these are factors of slowing down the production in organic farms. And insufficient supplies of transplants, which because not so many professional businesses just for producing organic transplants. So a lot of organic farms, they need to produce themselves, and that will, of course, gonna slow down, or the quality is not uniform. And the organic fertilizers, usually though, these are the ingredients are manure, compost, and animal byproduct, such as feather meals and bone meals and blood meals. And recently, there are also plant-based organic fertilizers, both in solid format and also in liquid format. And of course, there are liquid format of animal-based organic fertilizers, but those usually would have some smelling issues. Um, these fertilizers and in fertilizers and nutrients are usually in unmineralized forms that cannot be directly absorbed by plants. In open fields, there are soil microbes that those microbes would help to break down these organic matters and transform them into bioavailable form, mineral form. So the iron form. So for example, for nitrogen nutrition, the form that most plants would absorb is NO3 or NH4. Although some minor nitrogens will be absorbed through other forms like amino acid, but that's very minor. So why we are doing organic hydroponics? This is because hydroponics is growing in the US and around the globe, and greenhouse production is increasing as well. And greenhouse productions, many growers using hydroponics. US farmers are already growing a lot of organic vegetables in hydroponics and in substrate. And of course, the the biggest driving force is the National Organic Standard Board passed a decision to allow hydroponically grown produce to be certified as USDA organic. With that decision, um, it is expected that there will be more growers who want to grow organic food in hydroponics. Now, with the urban agricultural development, there are many production forms that production systems in urban areas can use hydroponics. So our objectives are to compare organic versus conventional fertilizers in two nutrient hydroponic systems. So one is nutrient film techniques, and the other one is deep water culture. And then we wanted to test the efficacy of organic inoculant. And although many those manufacturers of organic fertilizer, they would say, okay, our organic fertilizers can be sufficient without any supplement of other nutrients. But we wanted to know if it is true. So we did several studies. We also compared different substrate during the propagation stage. So we used the two lettuce cultivars. So one is the green ones and the other one is the red one. So both are organic lettuce. So we have three treatments, and we have the control using the conventional fertilizer. We use the whole ground solution, but we modified according to our needs. And the organic fertilizer we use is pre-emit. So and then we add one inoculant, which it is not a formal study, but other growers have used these combinations using Talabella, which is a bacteria-based root inoculant, mostly used for open field and also can be used for substrate. So it's basically soil microbes. So our hypothesis is if we add inoculant, inoculant can help release the organic fertilizer. So we we wanted to achieve if we can achieve the equivalent yield compared to conventionals. And this is our setup. So we use a rooftop greenhouse, newly built with excellent temperature control. However, we have a problem with the shade because it's attached to the roof, and some the the this side close to away from the building receives more light, and close to the building they receive less light. So we have two blocks, so six, and then this small greenhouse. So the maximum NFT system we can fit in is only six. So and we have three treatments. So basically not a complete. So we have to repeat over time many times. And here is our first trial. Usually the NFT, we can just supply the nutrient solution continuously 24 hours. And of course you can do intermittent if you wanted to save electricity. And then we started with continuous flow, and then in 2 weeks later, we found that the root system is not developing as healthy as the conventional ones in both organic treatment, the organic fertilizer with or without inoculant. And that's 20 days later, still not catching up. So but we did here is the results. We did notice that the organic fertilizer treatment produce dark leaves, and so this is also matching to our SPAT reading. SPAT reading is SPAT is relative chlorophyll content. So you can see that the organic treatment has higher SPAT readings in both cultivars. And yield, this is the yield is as expected is lower than the conventional. The green cultivar has higher yield than the red cultivar. And this is the plants. So you see the organic fertilizer with or without inoculant, they do not have very as good roots as the control. So for both cultivars, that's the same. By the way, this was conducted in February, the end of January to February. So the light was lower, like the DLI, average is 10 to 12 moles. And even with this is the green cultivar, this is the red cultivar, even the red cultivar, you rarely see the red color. So we wanted to repeat if we would see the same same thing. So we repeated the same treatment in April. That's and then nine days later, we checked the roots and we found the same thing. The organic fertilizer has poor root system. So, and then later we tried other things, for example, reduce the flow rate and filtration, but that didn't work. And we eventually decided to just give them 15 minutes per hour instead of continuous. And that 10 days later, you see the roots develop much better. So that's the problem. So we really, for the organic fertilizer, we have to use dry the plugs, because when you I showed the roots, when you propagate those seedlings, when you transplant the roots, most roots are still inside the plug. And with organic fertilizer, which is different from the conventional fertilizer, the organic fertilizer, they have a big molecule compounds, and those compounds would maybe they formed some biofilm, or because of the compounds, it's just their molecules are bigger, and then they basically occupy the plugs, and then the plugs would have less air space, which would reduce the dissolved oxygen. So here is the yield. The good news is this is the second cycle. By the way, for the second cycle, the nutrient solution for the control, we started with a fresh nutrient solution. And then for the organic treatment, we use the same organic fertilizer, but add, we top the tank with new solutions. So which basically we retain some of the old nutrients that, because the organic fertilizer takes time to release, so we wanted to use that. And also the inoculant, adding the probably inoculant takes some time to affect. So the second cycle, we see the yield is growth, promoted the growth. And dry weight has similar trend among treatment. And again, the green cultivar is a little, oh, that's similar. So now we know that because of the substrate, because of the dissolved oxygen problems, we wanted to try different substrate, because different substrate would probably, with the organic fertilizer, causing different problems. So we added GFI. GFI is organic certified, is approved organic plug, and C box here, close the bottom organic plug from Cocoa Core. So that is also organic, and Eripod is also approved product for organic propagation. So we have these four. Rockwood is not, but anyway, we added it because it's such a popular, most widely used propagation plugs. So we wanted to see how they're gonna perform. And the third cycle is in May. So DLI is much higher. Now in the propagation stage, we already encountered some problem. We propagated indoor in our propagation rack, but we're using the same management. So we've noticed the sea box actually need additional irrigation compared to the rockwood. So they are a little smaller when they transplanted. So by the first week, the end of first week, we noticed this phenomena. This is the control and organic and organic plus inoculant. In the organic treatment, the rockwood propagated plants did not look well. They have discoloration, and that is because we look at the root system. So we saw that's the root system. That's the problem. That's this. We are using only 15 minutes, so giving them 15 minutes per hour. So for rockwood, 15 minutes is too much. So really depending on the substrate. And by the end of second week, we noticed that with the rockwood, most plants already recovered. However, without the inoculant in the organic solution alone, most plants did not recover. So they are still struggling with that dissolved oxygen problem, which means, as you can see, this is the rockwood only organic fertilizer without inoculant. This is organic with inoculant. So with inoculant, when they start to work, they break down the big molecules of organic fertilizer, and that would create some airspace in the plugs that and help the plants grow. So this is the lineup of the treatment, and among the four, the best substrate is Jiffy, and they performed better than the CBOBs and also Elipod. So here is the results of yield in the third cycle. Jiffy organic is the best, and the differences among different substrate need a different irrigation management, especially during the both stage during the propagation and also during production. For example, substrate like rockwood, we would need to extend the interval of irrigate of the NFT flow, and Elipod is a little too short and also not close the substrate well enough, so that creates some problems. So in the next cycle, we dropped this one. So in the fourth cycle, we just wanted to repeat the third cycle, but drop the other one, the Elipod, because the size does not fit into the NFT. Instead, we're using the Oasis plug. Oasis is another organic propagation plug. So we wanted to see if Oasis would have the same problems like Rockwood because of the nature of the plug. So, and here it is. Actually, it is true and even worse than Rockwood. So the root did not develop as good as the Rockwood. So because we could not change individually on each channel since we're just using one pump. We just wanted to confirm if that problem happens or not. So among these four, as you can see, the Jiffy is the best, and Jiffy even the organic with inoculant and has a similar yield. So it is possible that we can achieve equivalent yield in organic treatment, depending on how you manage it. So here is the lineup of the photo. Now, cycle 5, which we wanted to repeat the results we have achieved so far. So we wanted to just repeat. And then we also did some tissue analysis and nutrient analysis. And that the last cycle in NFT is in June, from June to July. And here is the leaf mineral contents analysis in the cycle 4 and cycle 5. The NPK, and these are only, I'm presenting only the macro. The micro actually, no problem at all. The macro, some elements depending on elements, but the obvious one here is the potassium. So potassium, cycle 4, cycle 5, both had a lower in the organic treatment, which means we should have supplemented with potassium for nutrition. during the production, because even with inoculant, probably potassium supply is not matching the plant needs. And here is the phytochemical analysis results, anthocyanin and total phenolic content. Anthocyanin, this, as we observed, even from the first one, the organic fertilizer would give higher anthocyanin content, and also here it's not so obvious, but the SPAD reading is obvious, and also visually it is. But as the light levels increased, then the organic effect diminished. So that's the problem. And for the other ones, for the total phenolic content, which is a type of antioxidant, this actually, the organic fertilizer did not have significant effect, but the block has a significant effect, which means light treatment is much more obvious, or much, if you wanted to increase the phytochemical content using light treatment, is much faster than fertility fertilizer treatment. That you will hear another talk in this afternoon. Triston is going to give a talk on, on supplemental lighting, how we supplemented the light spectrum, and when you supplement, that gonna all make a differences. And then this is the final cycle yield. Although the organic did not achieve to the level of conventional, but we are confident that if we manage it well, it will be. And also in our studies, all the control treatment we started fresh, and the control treatment actually has pretty high EC levels, higher than normally growers are going to use. So we started with the 2.4 and then gradually reduce in summertime 1.8. So that's probably also a fact that the differences is so big between organic and conventional. Now we wanted to switch to deep water culture. In deep water culture, we actually used a different organic fertilizer because another company provided a product that we didn't have a chance to use in the NFT, and also because of the limited space, we couldn't do them together. So we rearranged our greenhouse, and we tried to squeeze these 12 NFT systems, try to close to the road side, I mean, the highlight intensity side as much as possible, but we still have the design is three blocks, and each treatment in each block, 3 treatment in each block is randomized. So we have 12 DWC, and same 3 treatment, but different organic fertilizer. We're using the same inoculant. So we wanted to see if this inoculant are gonna also help the other organic fertilizer. So here is the reservoir EC and pH in cycle 1 and cycle 2. As you can see that the pH in both cycle at the later stage swing much bigger than the initially. So when the plants grow bigger. And here is the results. So fresh weight and dry weight, and we see significant differences between block again. So even though the light we thought this time would be very small, the light actually the light difference is not as big as the NFT. Since the NFT we used almost the whole greenhouse, and this one we only using one corner of the greenhouse. And the cycle 1, cycle 2, the same tendency. The difference, the surprising thing is that in the inoculant in this organic fertilizer didn't help. Even in cycle 2, you see we didn't see visually the color, and we didn't see visually the plant performance as well. So and And here is the anthocyanin and total phenolic contents. And that actually, you see the organic fertilizer actually has a little higher total phenolic content here. But in the, oh, okay. Oh, here is the photo. So block 1, block 2, and block 3, and organic fertilizer plus inoculant. So visually, you can see that the plants in organic fertilizer were a little darker. And here is the nutrient solution analysis. Cycle 1, we analyze, send, these are the samples we send to commercial lab. Cycle 2, these are the end, at the end of the cycle. So in between, there was some problem with the commercial analysis lab. Since our organic fertilizer, they had a problem analyzing our solution. And initially we didn't tell them this is organic fertilizer. And then after they learned, oh, it is organic fertilizer, and then they treated it differently. And then they completely mess up our sample, so we didn't get results. The results just not, did not make sense, so we didn't present. And even this sample, we asked, one time we asked them to reanalyze, and they reanalyzed for some, but didn't give us a complete results. So that's why our nutrient solution analysis results is incomplete. But initially, as you can see, NPK and the nitrogen, they all have high levels. However, the phosphorus and the potassium, initially they started at a lower level compared to the conventional one. But we thought this is okay because this is organic fertilizer. They should release gradually. So we wanted to know how they change. So that was a challenge that we didn't, even though we paid, but we didn't get the results. And tissue analysis, these are good. So that doesn't matter if it's organic or not. So they're using the same method. And we found that cycle 1, cycle 2, that the potassium, leaf potassium is lower at the, so even though this is at the end of the cycle, potassium seems like not lower compared to the control. So I just could not figure out why. The assumption is that in between, they didn't release fast enough. So even though at the end of the cycle, the potassium in the tank is reached to the high level, but it's too late. So that's probably the only explanation I can have for this. So in conclusion, the yield in conventional is always higher than organic, but there's a tendency that in some treatments, that in some cycles that we were very close to achieve equivalent yield as the conventional. And the inoculant helped in one of the organic fertilizer, but not the other. The one that didn't help is the organic fertilizer look so different from the first one. So really, we need manufacturers to tell us more about the initial data so that we can target if we need to add magnesium or add potassium. So those are the things we really need to do analysis. The problem with organic fertilizers is you cannot just analyze one time, not like conventional ones. The organic fertilizer, because they are changing. So day 1 is different from day 2, and from week 1 or week 2. That's something challenging. So we have to be patient. So we do need to find out a standardized way to analyze those organic fertilizer. So the good news is that organic fertilizer increase the color of the leaf and increase the anthocyanin. And especially in winter time, it's more obvious. And in summertime, because the light level is very high, then the effects of organic fertilizer diminished a little bit. And the total phenolic contents are more influenced by the amount of light. So that's something if you wanted to increase the those phytochemicals, and using light treatment at the end of production is more effective than try to figure out using your nutrient solution. For the leaf mineral contents, they differ the amount treatment, but it depends on the elements. And when the leaf potassium is lower in organic plants in the 2 cycles that we analyzed, that indicated the needs of supplemental organic fertilizer of potassium. So that's what I have for today. So I would entertain for any question if we have enough time. Thank you so much for listening.
Yes, Dr. Niu, we have a couple of minutes. One question. Tristan has been helping answer questions in the chat message, but there's one question before we move on is asking, how did you optimize EC in the organic solutions?
Oh, the EC in this is our first project, so we basically follow the instruction from the manufacturers, and we wanted to have a comparable. First, we wanted to see if we can have a comparable EC, but the manufacturer says you cannot start with too high. Don't start with higher than 1.0. But later on, we try to increase. We actually increase in some of the cycles, but for really EC is not a good way to unless you know the composition, like this much EC and how much equivalent to how much nitrogen or how much NPK. That would make sense. I think we should match with NPK, and we really need to also know how this organic fertilizer gonna release, so that we can match the needs of the crop, and that which means the EC gonna change. And also EC also depending on what water you are using. If your water originally has some EC and the EC usually is contributed by all the macronutrients, and then the macronutrients does not mean all the macronutrients would be useful for the plant. For example, if you have a large amount, we still consider their macro amount like sodium chloride. Those are harmful ions. That if their ion concentration is high, that's going to increase the EC, but that's useless to the plants. So really needed to see the useful elements.
Okay. Okay, thank you. So if I understand you right, that and if I remember right, once we set up the EC for the organic solutions initially, per the recommendation of the label, we did not try to manage it or adjust it up or down during once the experiment was started, correct?
No, we didn't adjust according to EC.
Exactly. Okay. All right. Well, thank you, Dr. Niu. If you can stop sharing your screen.
Okay.
Okay, thank you, Dr. Masabni. Can you hear me?
I can see you, but I don't see your screen. If you can share your screen.
All right, can you see my presentation?
Yes, I see it, but not as a slideshow. Yes, perfect.
Okay, awesome. Thank you. Thank you for listening. Thank you for the introduction, Dr. Masabni. My name is Triston Hooks. And today I will be presenting one of our recent studies on the effect of supplemental pre-harvest UVA and red and blue LED light on lettuce quality. Okay, I'll start with a brief introduction. I'm sure this is familiar information, but greenhouse and hydroponic production of leafy greens is expanding globally. It's an important component of the world's food supply. And additionally, indoor controlled environment agriculture is a burgeoning industry, at least here in this area of the world. And it's expected to play a critical role in reinforcing food security in urban areas. And I think we've seen this hold true, especially in 2020 with the global pandemic and strains on traveling and shipping and transportation. But there are some issues. Both of these production methods typically block or completely lack UV light. A lot of greenhouse material, whether it's shade cloth or plastic screen or just the greenhouse material itself, can block UV light. We found in our own measurements that greenhouse material can typically block around 50 to up to 95% of UV light entering the greenhouse. And there is some evidence in the literature and that there's some potential benefits to supplementing with UV light. And so there may be, these production systems may be missing out on some potential benefits. And so we wanted to investigate this. Here's one example. This is an organic leafy lettuce. The variety is Red Mist. On the picture on the left-hand side is from actually a picture from the seed company. And this is their Red Mist lettuce grown outdoors in a field setting. And then in comparison, the picture on the right is the same variety of Red Mist lettuce, but it's grown in our greenhouse here. And there may be, and there certainly is, lots of factors contributing to this. The growing conditions, temperature, but certainly light quality and intensity plays a role in this. And we suspect that UV light plays a critical part in developing a pigmentation like this in lettuce. Also, we've heard reports from growers, local growers in the area, that they also struggle with developing coloration and pigmentation in a variety of crops. And so, again, UV light can be an important factor. Okay, so I also want to talk about, I want to talk about UV light, but before I talk about UV light, I'll briefly talk about just light spectrum. So we know that light spectrum can influence plant growth and quality. Here are just a couple examples. Red light, we know that red light is the primary driver for photosynthesis and growth. So we know red light is important. Also blue light, we know blue light's important, and blue light can increase pigmentation and phytonutrients. This has been shown in research. So these are kind of more obvious components of the light spectrum that we know about. But what's not as obvious is that light spectrum beyond visible light is also important for plant growth and quality. And I'll talk more about visible light, the visible light spectrum. But what I'm really referring to is PAR light, photosynthetically active radiation. But light spectrum beyond PAR, or beyond the visible light, is also important for plant growth and quality. For example, research has shown that far red light, which is beyond PAR light, can enhance photosynthesis when it's combined with PAR light, and that's because it has a synergistic effect. And this can lead to increased photosynthesis and increased leaf area expansion, and all of this can lead to increased growth. And then in regards to ultraviolet light or UV light, there has been some research that has shown that it can increase pigmentation of phytonutrients. But it's also, there's a cost to or a trade-off. It also has been shown to suppress or reduce growth. And I'll talk more about this in a minute. Okay, so here is a nice diagram that I found showing the electromagnetic spectrum. And I hope you guys can see my mouse, or I'll just try to talk. But it's showing light here as a wave. And on the left-hand side, there's longer wavelengths, and it goes all the way to the right-hand side with shorter wavelengths. And of course, you see on the far right, gamma rays are here. So the shorter wavelengths have a lot of ionizing energy, and they're harmful. At the far right here, they're harmful. But right in the middle here, we have the visible light spectrum, and it's enlarged here at the bottom. And so we're familiar with this. We got red light. This is the longest wave, 700 nanometers to 600. And then in the middle, you have yellow and green. And then on the far right, you have the blue spectrum from 500 to 400 nanometers. Okay, so this is, again, just a reminder, this is PAR light, photosynthetically active radiation. So plants perceive light similarly to the visible light spectrum. But as I talked about before, far red light is beyond the visible light spectrum. It's here in the infrared spectrum. So that's where far red light is. And then I'm going to focus on ultraviolet light here. This is on the right-hand side, so it's right beyond blue light. We have the ultraviolet light spectrum, and I will break down the UV light spectrum next. So UV light is broken down into three main categories. There's UVA light from 320 to about 400 nanometers. Then there's UVB light, which is from approximately 280 to 320, and UVC from about 100 to 280. So UVC light, this is your short wave UV light. And this type of UV light is the most harmful. It's used in a variety of applications for sterilizing or cleaning. And it can also cause skin cancer. So this is the most harmful UV light. We want to avoid this type of UV light when we're growing plants. But in the middle, we have UVB light. This is intermediate wavelength UV light. This has been used a lot in research. It's also responsible for giving you a tan when you're outside in the sunlight. So UVB light is not as harmful as UVC. And finally, we got UVA light, which is what I want to focus on for this presentation and for our research. UVA light is the least harmful. It's called long wave UV light. It's used in some applications. It's also known as black light. But this is the safest form of UV light, and we'll be focusing on this for the rest of the study. Okay, so like I mentioned in the last slide, research has primarily focused on UVB light. And I think one of the main reasons is because UVB light was engineered first, or easier to engineer, I think, especially for LED light units. But anyhow, research has primarily focused on UVB light. And they found that UVB light induces stress responses in plants, and they found a specific photoreceptor. called the UVR8 photoreceptor. And these stress responses lead to a variety of secondary metabolites that are produced in the plant, and they can increase pigmentation and phytonutrients in the plant. But again, like I said earlier, there's a trade-off. There's a suppression of growth because this UVB light is stress. It's perceived as stress by the plant. Okay, so what do we know about UVA light though? We know that UVA light is distinct from UVB light. And what I mean by that is it's perceived by different phytochromes. It's not perceived by that same UVR8 phytochrome that was identified for UVB light. Instead, it's perceived by cryptochromes, which are also the same, which are also perceived blue light. So they share the same phytochrome as blue light. And there has been some research that has shown that they can increase phytonutrients in microgreens without reducing growth. So there's some potential for UVA light to be beneficial and increase phytonutrients without reducing growth. So that's a good sign. Okay, and briefly, I've mentioned phytonutrients. So briefly, I wanted to describe phytonutrients. They are plant compounds that require light for biosynthesis. It includes a lot of things, including pigments, antioxidants, anthocyanins, carotenoids, flavonoids, just a lot of we also call them, they're also called secondary metabolites. You may have heard that term. So these secondary metabolites are also a component of phytonutrients. And these are beneficial to human physiology, health, and chronic disease prevention. There's a lot of research showing a lot of positive effects from these phytonutrients. So increasing phytonutrients in our produce that we're growing, this is beneficial. This is an increase in overall to the quality of the produce that we can grow. Okay, so putting all this information together, we hypothesized that we could supplement UVA light on our red lettuce production, and we could increase pigmentation, increase phytonutrients without reducing yield. So these were our hypotheses. So we conducted 2 replicated experiments in our greenhouse here. But before that, we did do a pilot test just to test this UVA light on our lettuce production, and we found that you need quite a lot. You can't have too little. So for our replicated experiments, we doubled up on the light intensity. I'll talk more about that later. But we increased the UVA intensity. We wanted to target the end of production. I call it EOP here in this presentation. It's the same as like a pre-harvest treatment. So we wanted to target that end of production time. We didn't want to provide the UVA light for the entire production cycle. We didn't think that was very practical. So we targeted the end of production, and I'll explain more of this in the next slide. But we also wanted to compare between day and night durations. So if we provide this UVA light during the day, is that going to be different than providing the UV light during the night? So we were trying to tackle a lot of questions at once. And also we used a red and blue light as a comparison. Okay, so here is a figure showing the supplemental EOP light treatments that we provided. We had a total of 6 of these light treatments here on the left-hand side of the figure. We also had a control. This control, I mean, there's no supplemental EOP light. So this control just received the natural sunlight that was entering our greenhouse. Okay, but let me break down these treatments. The top 3 in solid blue, these are the day treatments. So these were provided during the day. And then the bottom 3 in the blue shaded areas, these bottom 3 treatments were provided at night. And then at the bottom of the figure on the x-axis, we have the EOP days. So here's harvest. Here's the end of the experiment where we harvested. And then, for example, we provided 4 of our treatments at 2 days before harvesting, or 2 EOP days. And then here, we provided 2 treatments. These are UV treatments at 4 EOP days. So these were a longer EOP length. And then just to break down the names, how we named the treatment, this is our red and blue for 2 days, our UV for 2 days, UV for 4 days. And then again, red and blue for 2 nights, UV for 2 nights, and UV for 4 nights. Okay, in this figure, this is showing the same treatments. Here they are at the bottom, but this is showing the light intensity and the quality of these 6 treatments. So again, here's the control. There's no supplemental light treatments, but the natural DLI in the greenhouse was about 16 to 17. So we had a decent amount of natural sunlight in our greenhouse. Okay, but let's break down these 6 light treatments. On the left side of the figure is the intensity, or the photon flux density in micromoles per meter squared per second. And if you notice, all 6 of these treatments, we set them to have the same intensity of about 100 and 171 PFD. We wanted to do this because we wanted the intensity to be the same, because we wanted to really compare the difference in the quality of the light treatments. So here's the breakdown of each treatment. So here's the red and blue light. There was 96 PFD of blue and PFD of 75 of red. So there's actually more blue light than red in this RB light. And then for our UV lights, we measured them, and the breakdown, they only had a PFD of 41 UVA. And the rest was blue light, and that was actually a little surprising. So they had a lot of blue light in these UV treatments. So that's the breakdown of the light intensity and quality. Oh, and they provided a DLI of 7.4. So when you combine that with the natural DLI, these treatments had a pretty high DLI. So for the last 4 days or the last 2 days of production, these plants were receiving a higher DLI, of course, than the control. So in theory, we should see increased growth as long as the UV lights did not stress the plants out. Okay, so here is a picture of the UV light bar. This is provided by Fluence. So here's a picture on the right-hand side of the UV light bar that we used. And here's the spectrum, the breakdown of the spectrum from the UVA light bar, the figure on the top left, sorry. And so here's the peak is at 403 nanometers. So the peak is actually right on the border between UVA and blue light. And you can see, half of this peak is in the UVA range. And then actually a little more than half is in the blue range, from 400 to about 450. So there's the breakdown of the UVA light. And then on the bottom left figure, this is just the breakdown of the red and blue light that we use. This is also from Fluence. So this blue peak was at 450 nanometers and the red peak was at 665 nanometers. Okay, so here's a kind of a picture of how the experimental layout worked. We used kind of a shallow water culture system. We used these bins filled with nutrient solution. They're standing on a frame. And we had a floating raft on top to hold the lettuce. And we hung our lights. You can see here, we hung our lights right above the plants. Now, each of these systems was a light treatment. So you can see here, actually the control is on the far left here. It's kind of out of the picture. The control is over there. And then we have our, each of our light treatments has its own system. So let me show this. Okay, so for example, the 3 treatments here that are highlighted were our day treatments. They went from 7 a.m. to 7 p.m., so a whole 12 hours. And so you can see our red blue light here, we only needed one of them. The intensity was a lot stronger, so you actually had to raise it higher. And then our UV lights are here, we doubled them up, and we had to drop them pretty low to get a similar intensity. And then this picture, it's at nighttime, and it's highlighting our night treatments. So the same 3 treatments, but they ran during nighttime. So again, the red and blue and our 2 UV treatments. This one went for 2 nights and this one went for 4 nights. Okay, this is just a close-up. This was at the end of the experiment, right before harvest, but I just wanted to show we had these insulated reflective material, kind of like to be like light shields to prevent light pollution from one treatment to the next. So they were really effective. They worked really well. And we hung these up only during the last 4 and 2 days during the treatments. Okay, here are the greenhouse conditions during both experiments. This was done recently. This was done in September and October. Air temperature was very similar, about 23, 24 Celsius. Relative humidity, there's a little difference between the 2 experiments, 55 and 64. CO2 is very similar between the experiments, about 470 to 480 ppm. PAR was different. The light intensity that we measured, the natural sunlight in our greenhouse, was different between these 2 experiments. And we realized that our first experiment had a lot of overcast and rainy days. So this actually affected the results. And so I'll talk more about that later. So keep this in mind. But yeah, the end result, the DLI, was a lot lower, again, in this first experiment than the second one. Okay, so here are the results, the photos. This is from experiment 1, showing some representative plants from each treatment. So on the top left side is the control. And then I've got the RB, the red and blue light treatments here. And we've got UV light treatments for 2 days and nights. Okay, so the day is on the top and night is on the bottom. So we've got UV. And then on the far right, we've got the UV for 4 days and nights. And you can see, just based on this, the difference in the coloration and the pigmentation between the different treatments, and especially compared to the control. So we did see some increased pigmentation in our UV treatments. Maybe not as much as we were hoping for, but we did see some increased pigmentation, which was good. But kind of what stood out actually was this red-blue treatment at night seemed to increase the pigmentation the most, at least visually. And so we'll talk more about this later. This visual pigmentation that you can see correlated really well with some of the phytonutrients that we measured. But first, let me talk about growth. So this is, again, experiment 1. Okay, and the breakdown of the treatments here on the bottom of the figure, you got control here in the blue, and then we got the day treatments. I want to focus on the UV ones. And then here we got the night treatments. And again, I want to focus on the UV ones. But growth, these were great results. Growth was not suppressed. Here's the control. This is the shoot fresh weight up on the top. And our UV treatments, the day ones here, the green and the purple, and the night ones here, the teal and the pink. They were not suppressed compared to the control, which is a good sign. So UVA did not cause a lot of stress in these plants. In fact, we had a significant increase in yield, especially in this one. I want to highlight the UV for 4 nights. We had a significant increase from the control, but also the UV treatment for 4 days also significantly increased growth compared to the control. And then the same thing for the red and blue lights. And this kind of made sense. As long as these lights weren't stressing out the plants, they were adding additional intensity and additional DLI to the plants. And then shoot dry weight followed similar patterns. The biomass production of the lettuce followed similar patterns, and there are some significant increases compared to the control. So overall, this is good signs for the UV light that they were not stressing the plants out. Okay, now this next figure is showing the phytonutrients. So I'll break these down real fast. Up on the top, we've got carotenoids. And carotenoids as a pigment, are pigments. These are red and orange pigments. Beta carotene is a type of carotenoid. So this is a precursor to vitamin A. So this is a beneficial phytonutrient. Okay, next in the middle, we have anthocyanins. Anthocyanins is another pigment. These are blue or purple pigments. And anthocyanins also have antioxidant properties. So another beneficial phytonutrient. And then at the bottom, we've got TPC. This stands for total phenolic content. This is a very large category of plant compounds, but this also includes other antioxidants and beneficial phytonutrients. Okay, so what we found is that the UV light had some significant increases from the control in essentially all of these phytonutrients, which was really exciting to see. So I'll highlight this middle one. This is the UV light treatment for 4 days. We had the most carotenoids in this treatment and significant increases from the control. So that was very good to see. And then again, anthocyanin, this relates to kind of that pigmentation. And this, the greatest amount of anthocyanin was actually in this red and blue light treatment at night. And this is kind of the one we saw from the photos that looked like it had the deepest kind of red and purple colors. So this kind of matched really well with the photos and what we saw visually. But the UV light for 4 days also increased these anthocyanins compared to the control. And then finally, for the TPC, we also saw some significant increases in the UV light compared to the control. Okay, so just a summary of our experiment 1. The supplemental UVA light did increase pigmentation visually compared to the control, although not as effective as the RB light. And then we did not see a reduction in plant yield and biomass. In fact, we saw the opposite. We saw an increase. And then we also saw increased phytonutrients, which was great. We noticed that the 4 days of supplemental EOP UVA light was more effective than 2 days. So the 2 days just wasn't strong enough, at least at the intensity that we had. So the 4 days is more effective. And then the differences between the day and night treatments, we saw a little bit, but overall it wasn't very clear. But I'll talk more about this in experiment 2. The differences were more clear in experiment 2. Okay, so this is the photos from experiment 2. Again, remember the environmental conditions were slightly different. We had more natural light intensity and more DLI, natural DLI in our greenhouse conditions for experiment 2. But as you can see, the control still had very little pigmentation, and we had a lot better pigmentation in our light treatments. I think there is greater pigmentation in our UV light treatments and in the red and blue light treatments. Again, it looks like the red and blue light treatments increase pigmentation more than the UV. But we'll talk more about this in the figures next. Okay, so here's, this is showing plant growth. So we got shoot fresh weight on top and shoot dry weight on bottom. So we didn't have any significant differences actually in the shoot fresh weight. And this was different from the first experiment. And we kind of attribute this to the increased natural light intensity and DLI that the plants received in the greenhouse. So the control plants grew a lot bigger and a lot better. So the supplemental light treatments did not cause a significant increase. But what's better is that they did not cause a significant decrease. And that's really what we were hoping to see, is that they did not stress the plants out or decrease the growth. So that was good. And then again, shoot dry weight, no decreases. We actually did see a significant increase in one of the light treatments, the UV light treatment for 4 nights, had a significant increase in the biomass production. Okay, and then again, I want to show the phytonutrients for experiment 2. A little different from experiment 1, but again, we had significant differences in the treatments. I want to point out the one in the middle, the UV for 4 days. This performed better than the UV for 4 nights. We had significant differences here in TPC. So we had greater TPC content in the lettuce for the UV treatment at 4 days compared to 4 nights. So this is really interesting. We didn't quite see this for the other phytonutrients, carotenoids and anthocyanins, but we did see this for TPC. And this may indicate that the UV light is more effective when applied during the day than at night. And this can have some other implications with how UV light works with PAR light. Also to note, again, there were significant increases from the control in TPC, and I'm going to mainly focus on this UV for 4 days. Significant increases from the control, and also in anthocyanins, significant increases from the control. For carotenoids, there were actually no significant increases, but overall, there were some numerical increases that you can kind of see. Okay, so let me wrap this up in a summary for experiment 2. So again, the supplemental UVA light showed increased pigmentation visually compared to the control. Again, not as effective as the RB treatments. So I guess to explain this, blue light may be playing a big role here. So I'll talk more about this in our conclusions and kind of how we want to further pursue this and investigate this. But blue light may be playing a big role here. But we did not see any reduction in plant yield and biomass again. So this was really encouraging to see that UVA light appears to not induce any stress or at least suppress growth at the intensities that we used. But they did increase phytonutrients. So UVA light has some potential to increase the quality of at least lettuce. Okay, so again, the 4 days of the supplemental EOP UVA light was more effective than the 2 days. So we want at least 4 days at the intensity that we were using before harvesting to have a significant increase in the quality. And we did find that the UVA light was more effective at day compared to night. And this is mainly in that TPC phytonutrient, where we had a significant increase during the day than the night. And again, this has some interesting implications in how UVA light may interact with PAR light. But what was interesting is that the RB light seemed to be more effective at night to increase phytonutrients. And this may have to be that the RB light treatment is, during the day, the plants are getting the natural sunlight, and during the night, they're getting the red and blue light treatment. So they're just getting a lot of PAR light during the day and night. And so this increase in intensity in blue light overall just led to increase in the phytonutrients. Okay, in conclusion, we showed that the supplemental UVA light increased pigmentation and phytonutrients without suppressing growth in hydroponic lettuce production in a greenhouse. The supplemental UVA light was more effective during the day, and the higher UVA intensity may be possible without suppressing yield. And so what I'm trying to say here is that there's room to increase the UVA light even further. There's potential to have even more UVA light. Because we didn't see reductions in yield, we may be able to increase the UVA light even more and maybe even see even more increase in pigmentation and phytonutrients. So that's kind of a possibility and a direction that we may want to go. And of course, more research is definitely needed to better understand the effect of UVA light, especially on different species in cultivars. We just tried one variety of lettuce, but certainly whether this, we can increase the quality in other leafy greens or other crops. So definitely more research is needed and encouraged. Okay, I think here's some references, but that is all I had. So thank you very much for listening.
Thank you, Dr. Hooks. Dr. Niu has been answering the questions as they've been coming in the chat. I left one for you if you have a minute. The question is, do you think the control in the second experiment performed better because it is near to the better natural light as compared to the other treatments?
No, I don't. And let me go back real fast and show kind of a picture. Okay, how we arranged all of these, we get the most natural light from this side of the greenhouse where these windows are. This faces the west. We get a lot of natural light. So all of these in a row here, we arranged them in a row because they get similar natural sunlight. And of course, these shields are only up during the very end. So they're not up during the majority of the production. So no, we're pretty confident that most of the treatments received a very similar natural sunlight. Okay, I hope that answered the question.
Yes, thank you. Thank you. Thank you, Dr. Hooks, appreciate that.
All right, can you see my screen and hear me okay?
Yes, screen and audio perfect. Thank you.
Okay. Good morning. I hope you're all really doing great. And my name is Shu Yang, and I really appreciate this opportunity to talk to you today about how to efficiently manage lighting in your greenhouse or indoor vertical farm. So in the next 40 minutes or so, I'm going to look at why it is important that we have proper control of the light environment for crop production, and also what kind of approaches we can use to optimize lighting. More specifically, I hope to help you better understand how light affects the yield, quality, and market value of your crop And here in Texas, we get plenty of sunlight. In fact, there's often too much sunlight during summertime. So shading is relatively easy to implement and inexpensive. What I wanted to focus on today is to look at whether we need to provide supplemental lighting during winter time in your greenhouse, and also is it profitable to do so? And if you do run supplemental lighting, when should you provide your lights, and how much light should you provide? We'll also briefly discuss the things we should consider when selecting light fixtures for your facility. So first off, why do plants need light? Hopefully we all know that plants use light for photosynthesis. And this is a process where plants convert light energy into chemical energy that they can use to grow and synthesize biomass. So this is just like we humans eat food to get energy. So how fast plants grow is directly related to how much light they receive. And plants simply won't grow without light. And because light is so critically important, plants have also evolved. They have evolved to change the way they grow in response to their light environment. So one example is that stretching or rapid stem elongation that we see in many species when grown under low light. The transition from vegetative production to flowering is also affected by light signals. So those we generally refer to as changes in plant development. Let's see if I can advance. So in the previous slide, you may notice that I have photons in place of light, and just in case this is not a familiar term, I'm going to first get some terminologies out of the way. I know Dr. Hooks has already briefly touched on this. So the more familiar term light is a subset of electromagnetic radiation that travels in a wave-like pattern. So it's essentially a form of energy, and the distance between two consecutive peaks is called wavelengths. And we're interested in this because different wavelengths corresponds to different colors of light, and blue light has shorter wavelengths than red. And the general understanding is that light from 300 to 800 nanometer is biologically active. And we roughly divide this region into 4 categories of blue, green, red, far red, and also UV. And I'm going to talk about the spectral effects of those colors in a little bit. So another important property of light is that they actually come in discrete packets or particles that we call a photon. So for photosynthesis, it's actually the number of photons that matters. So the photon energy is inversely related to the wavelengths. So blue photon would have higher energy than red photon. But it's really for photosynthesis, what matters is the number of photons we receive, not the photon energy. But on the other hand, the energy of photons contained affects really the energy balance of the canopy, and it drives transpiration. So this graph shows the energy flux distribution of sunlight as a function of wavelengths. Almost all the output from sunlight is within 280 nanometers, that's a UV region, to 2800 nanometers, that's the infrared region, and a little bit less than half is in the visible light from 400 to 700 nanometer. And this also happens to be the range that most efficiently drives plant photosynthesis. And as I mentioned earlier, the biological active range is between 300 to 800 nanometers, so a little bit broader than the photosynthetically active radiation range. So how much light we receive in this region? On a bright summer day around solar noon, we get about 2000 macromole per square meter per second of photons. And this is an instantaneous measurement. What we really wanted to know is how much light cumulatively plants receive on daily basis. So we can add it up, and we get up to about 60 mole per square meter per day on the brightest day of the year. And we can also convert this to the photon energy that plants receive in terms of watt per square meter as instantaneous measurement of megajoule per square meter per day as a daily total. So you may also have seen units like foot candle or lux, but those are metrics developed according to the light sensitivity of human eyes. And for horticultural lighting, we really should not use those units. So we also get about 8% of UV and 34% of farad. So now hopefully, well, all experts in light measurements, and let's look at a map that shows the amount of light we get in different parts of the US at different times of the year. So if we look at the two extreme cases, that's the brightest months in June and the darkest months in December. So in December, the total amount of light we receive on daily basis, or the DLI, in the northern climate can be as low as 5 mole per square meter per day. And that's really not enough for most crops. And even in Texas, we get somewhere between 15 to 25 mole per square meter per day. And the recommended optimal daily light integral for lettuce, for example, is 17 mole per square meter per day. And for fruiting vegetables like tomato, cucumber, or sweet peppers, they typically need twice as much. So remember, the map we just saw is what we get outdoor in the field. Inside a greenhouse with glazing material and overhead structure, and also maybe hanging basket, the DLI can be easily reduced by half. So in many greenhouses, we typically need to provide supplemental lighting in order to increase photosynthesis, also improve the quality and yield of your crop. One effect we have with supplemental lighting is actually we can shorten the production cycle. So this can actually have a pretty big impact on the economics of your operation. So in recent years, there's also increasing interest in growing crop in indoor vertical farms near urban centers. And in this case, all the lights that plants needs for photosynthesis have to be provided by electric lights. So here I'm going to look at an example of the growth of lattice in a fully controlled indoor growth room with different amount of DLI provided by a white LED. And not surprisingly, lattice grown under low light, they really had very poor growth, and the growth increased with increasing amount of light they get. And we saw similar results with spinach. And this is a study that I did in a greenhouse during my PhD at University of Georgia. And you can tell that I did the study during the summertime because the DLI in the greenhouse I got was 29 mole per square meter per day. The main purpose of the study was actually to look at if acclimation of lattice to different light intensity would affect how efficiently they can use supplemental light. And it turned out plants grown under low light, they not only have poor growth, but they also could not use the provided light as efficiently compared to plants grown under high light. They are also more susceptible to photo damage induced under high light intensity. Another example is that the production of ornamental, like young plants or plugs or laners, often take place during the winter time in order to meet the market in the spring. So what happens is in northern climate, the DLI in the greenhouse can be as low as 1 or 2 mole per square meter per day. So a study done by Dr. Roberto Lopez from Michigan State University found that if you increase the DLI, the cuttings receive in the greenhouse, you can really improve the rooting quality of your laners. And you can also greatly shorten the days it takes to produce marketable laners. And this means you don't have to run heating in your greenhouse if you finish your crop early. It also means you can choose to have an extra crop if you want to. So this is another example of how supplemental lighting really improves the quality of young ornamental plants production. But the downside of using photosynthetic lighting is that it can be really expensive. So let's say you use high pressure sodium lamps and you have 1,500 lamps installed per hectare of production area, and you run your lights about 40 hours a day. So this provides an additional DLI of about 4.3 mole per square meter per day. And that's really not a whole lot. But if you run your lights for 180 days per year, that's about half of the year, and you pay 10 cents per kilowatt hour, you will end up paying $150,000 per hectare per year for running the lights. And to put this in perspective, the average annual farm gate value of a typical vegetable greenhouse is $700,000 per hectare of production. So just the electricity for lighting can be about 20% of the farm gate value. And the numbers are even higher in indoor vertical farms. So about 40 to 50% of the operating cost goes to electricity for lighting and air conditioning. And that's really a lot of money. And if we can reduce the cost of supplemental lighting or just sole source lighting in greenhouse or vertical farms, we can improve the profitability in those facilities. And so one way to do that is by using energy efficient lighting sources, and LEDs are currently the most efficient options for horticulture lighting. And the best LED fixtures can be about twice as efficient as high pressure sodiums, which is the conventional choice for supplemental lighting in the greenhouse. And the lights, the LEDs also keep getting more efficient and less expensive. The other benefit is that they are available in many different colors. So potentially we can actually create a spectrum that's most efficient for photosynthesis, or you can regulate plant shape or secondary metabolite production with LEDs. LEDs compared to high pressure sodium, they are also dimmable and they have a longer lifetime, so lower requirement for maintenance. So LEDs do generate heat, but the light itself does not get hot. That means we can actually place the light close to the canopy surface. So one example is we can use LEDs for intra-canopy lighting for tall crops like tomatoes or peppers, or in the example you see in the photo, that's hops. We can also use LEDs in stacked shelf system in indoor vertical farms. So what we have done in the past few years in terms of optimizing lighting is really try to develop lighting strategies that's based on how efficiently plants can use the provided light for photosynthesis. And also we adjust the way we provide light based on the value of the crop and also how much sunlight you have available in your greenhouse, because the amount of sunlight you receive really affects how efficiently plants can use the additional provided supplemental lighting. Specifically, we try to address the questions including when should we provide supplemental light and how much light we should provide? Do we need crop or location specific lighting strategies? And also we look at what's the optimal spectrum for crop yield and quantity. And we looked a lot on the edge of the spectrum in the far right and UV region. So we saw this earlier. So different crops really require different amount of light. What you probably didn't see earlier was when we grew lettuce under full sunlight during the summertime in Georgia, they actually got tip burn. So this is a disorder that lettuce plants often develop under high light and high temperature and low transpiration. So with shading, when we had a DLI of about 16, so this is about the optimal DLI for lettuce recommended by Cornell University. One thing you may have noticed is that the amount of plant growth we get is not linearly corresponding to the amount of light we receive. So plants grow under 29 mole of light compared to 16, the biomass did not double. And the reason is that if you look at how plant photosynthesis respond to the amount of light we provide, it's really not a linear response, especially in the highlight region. So if we provide supplemental lighting of, for example, 250 macromole, if we provide under dark or when the light is very low, you get a big increase in photosynthetic rate. But if you provide the same amount of light under a higher ambient level, you will get a smaller return in terms of additional plant growth. And the return would further diminish if you provide under full sunlight intensity. But different crops, they actually, they have different ability to use light more less efficiently. In this example, medical hemp can actually use highlight much more efficiently than a crop like lettuce adapted to lower or medium light level. So how efficiently they use light also reflects their light requirement. For a highlight requiring crop like hemp, the quality of the growth would be significantly affected or reduced if you grow those under low light. So with that information, because we know LEDs, they are dimmable and they really have fast on-off cycles. So we can use those for some smart lighting control system. So not a simple replacement of high pressure sodium. Dr. Mark Van Eersel at University of Georgia, who is my PhD advisor, in his laboratory, we developed a strategy called adaptive lighting with LEDs. So what it does is, for example, early in the day, if we don't get a lot of sunlight, a light sensor installed at the canopy level would take a measurement and then send a signal to a data logger. The data logger can then calculate how much light we should provide with our LEDs to reach a user-defined target light level. And then we can turn on the LEDs to get to the target. But say when sunlight gets more intense, the quantum sensor can measure very frequently and then send a signal and automatically dim the light. So this will make sure that we only provide light when plants can most efficiently use light for photosynthesis. And you can also set different target light levels based on the type of crops, their light requirement, and also your ambient light level in the greenhouse. And follow-up studies we have done in Dr. Van Eersel's lab have shown that we can actually grow high quality crop with less light input using this system. So I'm going to transition to the spectral effects of LEDs. And this is a summary slide made by Dr. Bruce Buckby in the crop physiology laboratory at Utah State. I did my postdoc for 3 years with Dr. Buckby before I came here. Dr. Hooks has also mentioned some of the effects, so I'm just going to have a very quick summary. So blue photons, they are photosynthetic, but the effects on plant morphology and development is that they inhibit like cell expansion that could be stem elongation or leaf expansion. And this is an example of medical hemp grown under same light intensity or DLI, but different fraction of blue photons, and you can see that plants grow under higher blue, they were shorter and more compact, and this might be a good thing. And green photons, we generally think, so there's a misconception that green photons do not drive photosynthesis efficiently. And most studies we have done in Bruce Buckby's lab and other laboratories around the world have shown that green photons actually can be used almost equally efficient for crop photosynthesis on a canopy level, because they can penetrate deeper into the leaf and the canopy and be used efficiently by the lower layers of leaf cells or bottom leaves of the canopy. But they also facilitate human vision. So this is a comparison of hemp grown under red and blue light on the right, and also with addition of green photons. So under the purple color, you really couldn't really tell the health of whether they experience like stress or if there's disease or pathogen issues present. And with green photons, we can really have a better assessment of the status of your crop. And red photons very efficiently drive photosynthesis. With far red photons, they do the opposite of blue photons, and they enhance cell expansion that could be leaf expansion in some species like lettuce that's shown here. Or they could induce stem elongation in some other species. So this is an example of lettuce grown under different fraction of blue photons, and you can see that higher fraction of blue will shrink the size of lettuce. As a result, so the canopy will be smaller and they will be less efficient at capturing photons for photosynthesis, and this would affect how much biomass they can accumulate. So when we double or almost triple the amount of light we provide to the plants, you can see higher light really induced higher production of protective pigments like anthocyanin in those lettuce, and potentially we may market those redder colored lettuce at higher market value because they're richer in antioxidant content. This is another example of lettuce grown with or without far red. We used two different background spectrum, a cool white and a rather blue spectrum. And we kept the total photon flux or total amount of light plants receive constant. And you can see by changing the spectrum and adding some far red, the canopy of lettuce really had faster expansion, and so they can capture more light for photosynthesis, and the biomass also increased by about 30% in both treatments with far red. But in species that typically require a high light level like cucumber or hemp or tomato, the addition of far red photons actually would cause excessive stem elongation, which is not necessarily a good thing. And this is a study done by Paul Kusuma, a PhD candidate at Utah State University. And he saw similar response under higher light intensity. So plants grew bigger, but the stem elongation response was basically the same. The reason is that So if we look at the spectrum of full sunlight, there's lots of PAR, there's some far-red photons above the 700 nanometer region, but because leaves or chlorophyll efficiently filter out photons in the PAR region, the 400 to 700 nanometer range for photosynthesis. And so in under canopy shade, there's relative enrichment of far red compared to the PAR region. So plants actually use this as a signal that function as shade detection. And when they thought they were under shade, they would want to either stretch to potentially reach the top of canopy and outcompete their neighbors. That typically happens with really like highlight requiring species or shade intolerant species. But for crops like lettuce, the leafy vegetables, they typically, instead of growing excessively tall, they use a different strategy. They expand their leaves instead and trying to capture as many photons as they can. So far, we've talked mainly just about light. So light is important. It really, if we change the light level, it affects how much resource we need in terms of like water or nutrient or CO2 uptake. But it's just important to keep in mind there are other factors that also affect plant growth and development. And just as a simple example, if we look at the CO2 response, so this is again medical hemp grown under very high light intensity. And so at ambient CO2 without enrichment in this particular study, we got about 32 micromole per square meter per second of photosynthetic rate. But if we enrich the CO2 level, we can easily double the rate of photosynthesis. And keep in mind that CO2 enrichment is relatively much cheaper than the cost of lighting. So, so the take home is that for efficient light management, we want to make sure that other growing factors are not limiting. You may also really consider to custom your lighting strategy based on what kind of crop you grow, their light requirement, the growth stage, and also the location. And supplemental lighting in a greenhouse is always used most efficiently under low light. So what we've seen repeatedly is supplemental lighting will increase crop growth and quality, but because of the high capital investment in setting up your lighting system, it may not necessarily increase the near-term profit. What I wanted to leave you with is a very helpful web page that you can look at that may help you to develop more efficient lighting strategy for your greenhouse or control environment. So this is a USDA project led by Dr. Mark Van Ursel. It's called Lighting Approaches to Maximum Profit. And it has a pretty, so the web link is right there. It has a pretty good outreach page that aims to help you answer questions like, do you need supplemental lighting for your operation? Is it profitable? So we have a team of plant physiologists economists and engineers that work together to understand how crops respond to light spectrum intensity, photo period, and how it changes their physiology, how efficiently they use light, their light capture. And we are also trying to develop a low-cost controller that can implement the optimized lighting strategies that we develop from this project. This is just an additional resource that you can use to help you make decisions in terms of lighting management. And that's all I wanted to share with you today. And I wanted to thank you for listening, and also thank you, Dr. Niu and Dr. Joe Masabni, for inviting me to give a talk. And if you have questions, I'm happy to answer them.
Thank you, Dr. Zhen. This is Joe Masabni again. We had some questions, but Dr. Leskovar and Dr. Niu were taking care of them while you were presenting. If you can stop sharing your screen. Excellent. But I can see it, but you're still muted. So please unmute yours or. So unmute yourself so we can hear you. Let's see.
Okay, I unmuted. You hear me?
I can hear you and I can see your computer screen. Just start it in a slideshow format, please.
Okay.
You know, like press F5. That's the easiest way.
Okay, so did y'all see the PowerPoint?
Yes, I do. But can you start the slideshow?
Yeah, I'm on. Oh, let's see here.
So press F5 is the easiest way.
F5.
Huh. Right. Or go to slideshow on the top bar. Slideshow. Why don't you click on enable editing? Maybe that's what's
There we go.
blocking.
Let me try this right here. See if this works. Okay, does it show now?
You may be sharing the wrong monitor because we still see. Do you have two monitors?
Let's see here. Because on the screen here, iPhone Basic Advanced, I click this and I click share on this one.
Tell you what, click on enable editing first. You see that on the top middle. Click on that. There you go, click that. And now click on slideshow on top, on the menu bar on top, click on slideshow. Yeah, keep going right, top
Right
bar.
here?
Yes, but go right, you see slide. No, not there. Slideshow. No, go back. Keep going, not home, keep going right, right. There you go, slideshow.
Okay.
And then click from beginning. There we go.
Okay.
Thank you, James.
Okay, are we good?
We're good. We can hear you
Okay,
and we
cool.
can see it.
Okay, excellent.
Thank you.
Okay, so yeah, graduated from North Texas in 2007, specialty fitness equipment industry for 9 years. National Academy of Sports Medicine, certified personal trainer. And really the backyard gardening started in, I guess, yeah, really started taking interest was around 2010, 2011, when I saw my brother and his wife start harvesting stuff out of the garden. Since I was in the fitness equipment industry, that was definitely a part of my lifestyle with what I wanted to do. So it really, farming was never in my background. I mean, my grandfather, he did farm, so we visited his farm. I was on his farm, but we didn't really do much in terms of actually the farming activity. So when I say YouTube University, it was just that once I started taking interest in that, I mean, that's just what I did, was just spend hours and hours of researching. It was just something that really fascinated me. And reading books, like some of the books from Jeff Lowenfeld's Organic Gardening, teaming with microbes, teaming with nutrients, and permaculture, regenerative agriculture, and just all things agriculture related. I really didn't know too much about microgreens until 2016, but this is to kind of give you an idea about what my first rack looked like in my kitchen. When I started out, I heard about the microgreens in 2014, 2015, but I was really more focused on learning outdoor systems before I started getting into indoor systems. And I have to say, the speakers thus far have been really, really impressive. So I'm learning a lot from y'all. This is really going to be helpful in our transition when we outgrow our current location. We're currently in Hempstead. So this is really going to be helpful when we design and plan our next out location. So thank you for all the information today, everybody that's been participating. The biggest thing that interested me on the micros was, from a nutritional standpoint, the fast turnaround time, ability to grow indoors, small footprint, most interested me as a hobby initially. And it wasn't until the summer of 2016 when I started experimenting with microgreens, and it was very, very frustrating initially, because just the learning, while they do grow fast, there wasn't a lot of information out there on the growth side of things at that time that I was able to come across, only a little bit. And I'll get into that a little bit shortly. Initial investment, wire rack from Home Depot, T5 lights, they were the sun blasters, soil from hydroponic store, seeds from microgreen seed suppliers. Approximate cost per rack outfitted with lights, fans, grow medium with seeds was about $1,000. Now, for anybody else, the barrier of entry with microgreens, this is what makes it really attractive. It's very, very inexpensive. There's a growers that can start out with less than $500 and get something going. So we started with about 8 crops, currently growing over 90 right now, including edible flowers. The 8 initial crops were sunflower, radishes, cabbage, broccoli, pea, and various salad mixes, bean mixes. And one of the, the issues is when deciding which way to go is soil versus hydroponics or growing microgreens. What are the pros and cons? Well, one of the things that we found with when you have a good soil formula is that the diversity of growing microgreens is a little bit easier to expand. Like, for example, with sunflower and peas, we found that we tend to get a little bit more consistent results with that soil medium with respect to how the roots are able to take hold and to grow. It's not that the hydroponics doesn't work, they do. It's just you're really going to have to get that the nutrient levels dialed in. It's not that microgreens can't grow to the microgreen stage without it. It's just that what we found is that it really does impact the flavor profile. Even though there's kind of a general level of understanding that microgreens have within the seed enough energy to get to that microgreen stage. That's true, but especially when it comes to the chefs that we partner with, they really do notice the difference in as far as how that crop tastes based on its growing condition. So we've decided to stay with the soil on our main approach for production because it's easier for us to really maintain that consistency. I've seen other growers do a phenomenal job with hydroponically growing them in hemp mats, cocoa coir fiber, and they get great results. It's once you find that method, you get that dialed in, you just kind of stay with that. So this is the idea of what the first rack looked like. I didn't know what I was doing initially, but eventually just figured out after many, many failures, what works and what doesn't work. The thing is like I have here, very little information on how to on YouTube at the time, but found about other growers and courses through farming podcasts. podcasts that dealt with permaculture and other types of agriculture type of podcasts. That was really helpful. Bought courses online. Took 6 months for trial and error to really hone methods down. First online instruction was a PDF, but came across another December 2016 from a grower in Canada, and that was urbanmicro.ca, Chris Thoreau. That was more focused on the technical aspects of growing, business applications, video, how-to instructions. And I did not really consider the business possibility until after completing the online course, because initially it was just as a hobby. And so one of the challenges is market research. Who are my clients? How many growers and farmers currently sell microgreens, and to whom are they sold to? And so what I found is there were quite a few. There's several different big growers at the time, but now it's the landscape's completely different right now with respect to how many other companies are really jumping into the microgreens right now. It's really interesting to see the evolution of this in over just a matter of a few years. Beginning point, farmers markets, local chefs, local magazines, social media, generating the courage to approach restaurants. That was one of the biggest hurdles because I had no experience in the restaurant industry. I had no experience in talking with chefs. The biggest thing that you're going to find with the restaurant is consistent product. Do you have this? And that comes down to seed densities. Are those dialed in? The growth cycle time, the yield per tray. What you're going to find is with different seed densities, that really impacts the development of the canopy with respect to the leaf. What we find is that some chefs want that microgreen to have a specific size. So you can grow the microgreen size, or you can grow to the petite size. And that's what really is going to separate your average hobby grower to a grower that really starts getting a lot of referrals from chefs is that customization, that specific size that chefs want. Even sometimes the textures can vary from the size of that microgreen. To sell live or cut, that's a big question that we see in the microgreen community, especially those who are starting out. If you do live, how are you going to transport that? The trays that are live, typically those are 1020s, the 10 by 20 trays. So that's another challenge that has to be considered if you do live. Now, some growers, I've seen them actually, they will put the live trays in bread pans. Like when bread companies deliver their breads, they put them in those type of pans. Or do you do all harvested? So you have to think about the refrigeration aspect on coolers, or if you have a refrigerated vehicle to transport that. And we do both. We found that a lot of chefs, while they do like harvested, we find that the shelf life is going to vary. We find we get better shelf life on certain varieties. With the restaurants, like our baselos, we find we get way better shelf life live. And so that really comes down to communicating with the restaurant or the chef as far as proper care and maintenance. So I'll get to my screen next as far as how we actually transport ours. We actually built custom racks. Pricing, that's another big thing. How do you even price? Where do you even start? That's one of the biggest challenges that we came across. How do you even start our pricing? You can start out by going to different retailers to see where they're at. But the thing is that some of them are at such a large scale that pricing at a low price is going to be hard to try to overcome your own costs when you're starting up and you're just starting out. So that has to be taken into consideration. Your time to do the production, your time to transport. There's a lot of different costs that have to be factored into that labeling. Harvesting time, all those little nuances have to be factored in with how we price these to farmers markets, restaurants, co-ops, or online sales. The biggest thing also when starting out is crop failures due to environmental conditions, mold, improper seed densities. And what I can tell you is that when I started out in my house, when we started taking increased orders, we actually expanded into a guest bedroom, and outgrew that one. We ended up having, I had a small 1,200 square foot house and turned the whole thing into a whole entire grow house, pretty much. I mean, it was an interesting learning experience to see how crops react to different environmental conditions. You know, especially due to the perspiration and the humidity. And we had extension cords going throughout the whole entire house because we were popping circuit breakers left and right. So we had to map it out from one wire cord going to one side of the house, going to the other, alternating the lights when they're coming on. So it wasn't the ideal environment to grow, but it really got to show you how to, what to do, what not to do. When we moved to our next location, that's when things really started to get more of a commercial scale. And we'll get to that in just a second here. Customer order fluctuation. That's a real challenge sometimes when it comes to the order fluctuations with the restaurants. Traffic goes up and down. So you can actually set growth cycles a certain way that you set, say you do like a radish 8 days out, you have it set with the restaurant, and then say they, you know, they have a really slow week. What are you going to do with that radish? Well, the chef's going to say, well, we can't take it. So that's where it comes down to that balance. You know, if you have a standing order, they have to take it. But the thing is there's that balance of if you be really strict about it, is that going to be an issue with maintaining the consistency with your customer base? So having a little flexibility and alternative vectors as far as how to move product in case a certain customer is unable to take it is something that any growers are going to have to consider. And that's where farmers markets can be handy. That's where we move a lot of our excess, our surplus production. So the order fluctuation is going to be always an ongoing battle with any type of grower. So this is an example of our vehicle. And we're actually going to be getting a commercial vehicle pretty soon here with fabricated shelves in it, and it will be refrigerated. So, but from a do-it-yourself standpoint, and before this thing, I just had rubber mats on the floor, so that way the trays wouldn't even slide around. So, but now we have a little bit more structure. But this is kind of give you an idea. Right now, we're finding out that while we still do deliver a lot of live, especially after COVID, we're doing a lot of harvested varieties too. So we have, while we have that shelf right there, we have about usually 3 to 4 different coolers to store all the harvested varieties, and we make our deliveries. Time management, full-time or part-time grower. Let's see, let me get this. So a lot of growers, when they enter this, they already have a full-time job. The kind of the routine that I had to go through was when I was in that fitness equipment industry, I was working usually 5-6 days a week. So how to actually manage that time with meeting new potential clients, with maintaining the drive to meet new clients and to keep up the production. So, when I put drive to go full time with little to no sleep, that's a reality. When you start going full time, or even when you have a full time and you're trying to do this, you're going to be losing a lot of sleep. You're going to be realizing that you're going to be working for less than minimum wage. A lot of growers like to get into this thinking that this is going to be extremely profitable, which it can be, but the initial startup is when you're trying to build the accounts, and if you have already a full-time job and you're working around the clock, it's not easy. So when you're walking around like a zombie every day, like I was, it's a big challenge to just want to drive through that. I remember going to Whole Foods every single day and getting my daily dose of caffeinated drinks and chocolate to maintain my sanity. So it was dry caffeine and chocolate. That was my recipe to get through that time frame when things really started to pick up when I had the full-time job. And then when I went full-time growing, it's like it didn't stop because we had other issues that we had to take care of. So my routine before meeting with chefs was a lot of pre-workout supplements or even caffeine pills. So I'm not on that same routine, thankfully, right now, but it's not something that I wanted to do because it really took a toll on my health and my stress levels. But you have to think that, you know, when I was in my early mids, when I was, what, 33 at that time, I'm 37 now, this is my one shot that I had to do it, and just suck up and just move forward with it. And so when you're going through that kind of grind, I definitely recommend that take power naps, especially so that way, if you're avoiding accidents, there's quite a few times when you start thinking that you're dozing off the side of the road and you just have to go off to the side of a parking lot, take a power nap there, because at that time I was still operating off of 2 to 4 hours of sleep for around 4 to 5 months straight. Not to say everybody's got to go through this transition, but this is just the kind of grind that I had to go through. Making the transition to go full time, reality sinks in when you go full time 7-day work week. It doesn't mean that it gets lighter, it just means you have more work, because with microgreens, when you start, when we were manually irrigating 110 to 120 trays per day, you have to monitor every single detail of those trays with respect to when they're germinating, if they're molding out, when they need to be uncovered. So even though the microgreens are very fun, there's a lot of you know, they turn over fast. It's just the maintenance side of it. It's way more uptake than just growing full mature crop, because, you know, in 4 weeks you got 1,000 crops all ready to go. With microgreens, it's a daily maintenance and a monitoring thing, especially when you really start moving a lot of volume. Getting turned down at restaurants, self-doubt, questioning yourself if this line is right for you, if this line of work is right for you. That's one of the things that a lot of growers are going to have to overcome. Not all restaurants are going to want to use microgreens. Some of them, they're more focused on getting for the lower price point that they can get from some of the other distributors. But the thing is that you really have to have a game plan to show how your product has either better shelf life, better flavor, could be the presentation. There could be a variety of reasons that you have to really show how you can separate yourself from other growers or distributors. It could be a custom mix that you do, for example. Let's see here. Yeah, outgrowing the current growth rates, kitchen, guest bedroom, living room. Infrastructure costs continue during expansion with new rack, lights, timer, fans, and dehumidifier. Overloading the AC with crop perspiration, temperature from lights, humidity levels, not optimal growing conditions. Yeah, I mean, I can't tell you, I mean, we really overloaded that AC in that house with the amount of perspiration that was going on. It was a higher electricity bills as more lights installed, because with those T5s, they end up producing a lot of heat. So that house got pretty toasty during certain times throughout the year. What do you know, neighbors think of grow lights blasting through the windows? They thought we were growing, some may think we were growing cannabis. So we covered the windows with cardboard so our grow lights wouldn't be blasting out of the side at night. Circuit breakers popping. We had to map out circuit breaker outlets with extension cords so lights pull from different outlets. You know, and that, yeah, I mean, that was, that was quite an experience to outgrow that area. Transition to second location, how to scale self-automated irrigation and light systems to enhance workflow efficiency, layout design, production, harvesting, when to hire. The thing is with that is, this is what our current setup looks like right now. We have 4 pallet racks like this. We have 115-gallon reservoirs. We do not use nutrients in the reservoirs because with the soil formers that we have, we found that we really don't need it. The airflow, we use wall-mounted ceiling fans from Air King, and we use those as our main air circulation. Those lights there, those are T5 lights. It's a multi-bulb fixture. The thing is with those, we found that at that kind of distance, the 8-bulb fixtures were too powerful. They were so intense that they actually stunted the growth of a lot of the microgreens. So we had to pull 4 of those tubes out to reduce the intensity. And that's where we got the actual growth that we needed to. And those lights, we ended up getting a much better results than the standard T5s that we were using in the previous location. We got better colors, better stem structures. As far as the timing is concerned, we ended up using timers from Apollo to control all of our pumps and lights. The problem with the lights running through your lighting systems through those Apollo timers that we blew through around 13 to 15 of those, and they just froze. It was just too much juice. So we ended up getting an extra 200 amps installed into the grow house. So we had the HVAC separated from the actual, what the lights were operating off of, because we were blowing that circuit breaker all the time. So when we did that, we ended up actually utilizing a system that manages timing for pumps for pools, and used that to control all of our light timing. And we have had very little issues with that. So that transition has helped tremendously. No circuit breaker pops. And that was a fairly inexpensive approach, getting that installed instead of going with the, well, I wouldn't say the actual getting the 200 amps is inexpensive, but there's other systems out there which do control lighting, which do great jobs, but we had to look at it more of a budget approach. So that's why we utilized a control panel that controls pumps for pools and adapted that into the lighting system we currently use. The Apollo timers, we still utilize those on the pumps. And the biggest challenge with that is figuring out which pump to use per level. When I talked to the hydroponic company, they didn't even know what kind of, they gave us some relative guidance on what kind of pumps we could use. But to try to figure out for the bottom tray, for example, you know, the level 1, level 2, how long do we need to flood each tray for? Because you have to think that those pumps, when they're going up, gravity is going to impact on, have an impact on the flooding part. So some levels are 4 minutes, some are at 5, some are at 6. And then also some crops, while they get irrigated 24 hours a day, every 24 hours, some crops don't like it that much. Some crops like it every 27 or 30 hours. So having to stagger those timing, that was an interesting learning curve. And that's also going to be tied to your air circulation, intimately tied to your air circulation. So that air circulation, if you have a lot of air circulation in certain areas, some of those trays can actually dry out faster, which means that you may have to increase the irrigation cycle. Let's see here, we got that. How many hours are you working? Are you able to see new clients? Let's see where to hire. Yeah, Indeed.com was definitely a huge, huge plus for us to actually hire additional personnel. I mean, before I had additional help, even in this second location, I mean, I was still pulling a lot of hours. I was even sleeping on the floor. Take a 20, 30-minute nap at 2 o'clock in the morning, get back up, hit production, then get ready for deliveries the next day. I mean, that was not a fun routine, but that's what had to be done. But Indeed.com for any new growers, unless you know some people that are interested, that's been a huge, huge help for us to find potential people to come on and to train them. Then once you end up getting the new people on your team, that's when you're able to see new clients. So delegation, that's been a huge, huge learning curve, because initially I started out, I didn't really know just that business side, and delegating, that was sort of new to me. But the power of delegation really, really comes into play when it comes to building a business like this, because when you start out, you are the face of the company. And so you have to continue to see new clients and maintain those relationships. And if you're tied down to doing the production and all the nuances in the background, it's going to be harder to see new clients and to grow that business. COVID-19 pivot. So majority of our business was restaurant focused. That dried up when things shut down in March. That was probably that was most of our business at that point. We only had a small fraction of retail at the time that we were starting to get into. And we donated over $7,000 of products and donation spots, so product would not go to waste. We built relationships with retail, farmers markets, co-ops, online sales, and distributors. And that kind of pivot has allowed us to not rely so heavily on restaurants. Now things have opened up a bit. So restaurants have definitely come back online. That is still a core of our business, but we are growing these other areas. But the biggest take home I can tell any grower or any other business, especially in this type of specialized area, you really can't have your eggs all in one basket, especially with what's going on right now. You really have to think of alternative vectors to diversify as much as possible. And even with value added, we have a chef on our team. So now we're developing microgreen pestos, dressings, infused honeys. We're doing many, many different types of value added items. It's really exciting. The collaborations that we're doing with other businesses and utilizing the microgreens to really showcase how they can be utilized other than just as a garnish at a restaurant. What I wish was available to help me learn information on crops, which can be more sensitive to densities and growing conditions, cilantro, basil, and other specialty herbs. There was not a lot of information at the time when I was learning on how to do that. There's a lot more information right now on YouTube on growing that. Cilantro, basil, they can be a bit of a challenge on the growing side of things, especially when it comes to humidity. We try to keep our humidity at about ideally around 55%, but sometimes when we have rain outside or other type of varying conditions, even the outside humidity and temperature impacts the inside. So this is also why any growers, they have to also consider incorporating auxiliary AC and or dehumidifier units to control that. When our humidity starts going above 70%, that's when we start having a lot more mold issues with respect to cilantro and basils. So the extra dehumidification is going to be absolutely critical to any new grower, especially with microgreens because of the sensitivity of the crop. Production schedule templates to meet customer demand. Most farming software is for traditional outdoor farming, not indoor specialty industries like microgreens. There are some companies out there I've come across that do have that capability, but for small mid-sized growers, the price point to obtain that type of software can be a bit of a hurdle. So there are some grassroots apps right now that are starting to develop and softwares for small to mid-sized growers that are developing right now. And we're actually going to be beta testing some pretty soon here that we're really excited about. And some of these will have the capability for us to tie into NOP standards in terms of what NOP requires for organic certification. Understanding language used by culinary and beverage professionals, how to engage with meeting for the first time, what questions to ask. So when I say language, when it comes to the chef community and even the craft cocktail side, language is like when I remember when I went to my first restaurant, when I had that meeting set up, the first word that the chef wanted to use on the radish that we were introducing, he wanted to use it as a feature. So when I think feature, I had no culinary experience. I didn't know really what that meant. And so when I heard it being used in that context, I had to sit back and think about that in relation to how microgreens have typically been utilized as a garnish. So when it's as a garnish, it's not being utilized as an actual feature. It's just there just for decoration, for presentation. So when chefs usually use language like components, accents, features, they're thinking of when the chef designs a dish, they're thinking in their mind when they bite down, what's the first thing I taste? What's the lingering taste that follows at the end? What does the texture think? I mean, some chefs, it's almost like a, some of them I've actually come across, they'll actually close their eyes and they'll think about the colors that are coming in as they're biting down on that dish. They're really trying to immerse themselves in that flavor. So it gives them inspiration and ideas about how to really design that dish. These guys, the men and women in the culinary industry, the way the evolution has come is just absolutely, it's been a huge eye opener for me with how the thought process that goes into designing dishes and even the craft cocktail side of things. So really understanding what questions to ask. Like when they say, hey, my primary components are, it's a steak, it's a potato, or it's like if it's a plant-based dish, it's a carrot. I need to accent it with these type of spices. What kind of microgreens can I incorporate with this? I'm like, okay, that has that kind of spice. You could utilize this, but this might be too dominant on this flavor profile. So we may have to balance it out with this. So it's really you're formulating a dish based on the accents and the spices they're using with which you're growing. So you really have to know what your flavor profiles are on everything that you grow. in order to really help design those dishes. And with the craft cocktail side of things, we'll have bartenders utilize them in simple syrups. We actually had one that incorporated some of what we grow into a Bloody Mary. It's the best tasting Bloody Mary that I've ever had in my life. And I'm not a Bloody Mary person, but it was good. And what's interesting about the microgreens is that they macerate so easily. In the craft cocktail applications, you can impart those flavors so easily in so many different applications. And that's one of the biggest things that we really try to communicate with anybody is the diversity and how they can be incorporated. It's not just as a garnish. Years ago, they were referred to as just vegetable confetti, but the culinary trends are really shifting into utilizing microgreens in a capacity that historically has been relegated to only as presentation. So we have some chefs that are utilizing them into dressings. Like they'll actually harvest the leaf on some, like on a nasturtium, and then utilize the stem for the flavor into a vinaigrette. We've had some actually puree some of the stuff that we use onto some of their dishes. So it's really interesting to see how the evolution is being utilized in the food trend scene. The chefs largely impact the current trends we see in food. So when we see the chef community starting to engage in these types of areas, I think this is an area that all farmers and growers, if they take notice in this, it's really going to assist anybody that works with the chef community and ability to tie into that trend, that the evolution of the culinary scene. Let's see here, market availability, impact of certification. Some markets require certification of various degrees, such as GAP and organic. When it comes to certain distributors, they require third-party audits. So for any type of beginning or mid-sized grower, if they don't have those organic those certifications, trying to tap into those revenue streams can be a bit of a challenge unless you get those types of certifications or audits. Let's see here. What keeps you in the microgreen business? Microgreens are not a replacement of full mature crops that meet dietary needs, but complement to nutritional and flavor gaps for consumers and culinary professionals. And so even though we specialize in microgreens, we're not, advocating here that it's going to replace full mature crops. It's just that it can be understood as a complement. When we go to farm markets, people ask us, well, what are microgreens? It's the next stage of growth after sprouts. It's that language, and to communicate that to the consumer is really important. And so there seems to be a lot of equivocation with sprouts and microgreens. They are not the same, but they get understood as such. So making that distinction to consumers is really important. So they understand what they are, how they're different, and how they can be incorporated. The value added, the micro pestos that we've done. We've also done collaborations with sorbet companies. So we did a key lime micro cilantro sorbet, and it was out of this world. What's really interesting is that when was introduced that sorbet with mature cilantro, it was always a really good seller. But when we incorporated that micro cilantro, the cilantro came in way cleaner. Even the customer feedback was really interesting because they noticed way more cilantro in that sorbet. It's really interesting to see how, and when the microgreens are grown properly, that's when you can really showcase the flavor profile. That a little bit of that microgreen goes a long way. So when we use our micro basil into some of our tomato sauces or pestos, for example, we don't need a lot of that basil to let that basil really showcase into that. And that's the thing, if this kind of ties back into how it's growing relative to the grow medium, soil, hydroponic, nutrients, and the light spectrum. That's if it's not growing in the optimal condition, it's going to be a different scenario to really try to showcase the flavor profiles in some of those value added items, or with the chefs if the flavor is really not going to drive through. And we from our experience, we found that they seem to appeal to kids more than mature crops. And the case in point is the Woodland Prep School that we collaborated with. before the shutdown, we brought samples there, and we showed the second and third graders how to grow the microgreens. And what was really interesting is to see a lot of kids who didn't even like vegetables would come back for seconds and thirds from microgreens. And it really surprised the staff that these kids who normally don't go to lunch and eat vegetables are coming back for seconds, thirds from microgreens. And I think because they're so much smaller, they're unique. It's got that unique and that curiosity factor of what is this thing? It's different, I haven't seen before. That seemed to impact their decision or their interest in wanting to try microgreens. And so what it seems to me that if we're seeing that kind of reaction with kids at that age, and we're trying to incorporate nutrient-dense crops or getting more greens in their diets, for any parent that finds it challenging to incorporate greens into their kids' diets, I think micros might be your stealth bomber to get it into the diets, because they're so convenient. You just harvest, you put them on anything, whether it's a sandwich or soup or whatever your child likes. And it's not like a big stalk of asparagus. I like asparagus, but not all kids do. So I think the microgreens, from that perspective, it's something that we really need to communicate with parents, especially when they're trying to get kids healthier diets. I'd like to pull up this other screen here. How do I minimize this here and show?
Yes, yes.
Okay.
Press and
I want to
show.
I want to show some additional pictures here.
Sure. Yeah. Click and show to close the PowerPoint presentation.
Okay.
And then you can minimize the screen and open your other screen.
Okay. So this will give you this right here is what our package looked like when everything shut down on when we donated. I mean, it was just a ton of surplus. We did not want it to go to waste. So this is kind of, we sent this is one rack, and we have multiple racks. So they're just going out to different donations that we donated to different complimentary care packages for Houston restaurant and first responder communities. Some of the images to kind of see how the chefs, when they plate. This right here is an example of an nasturtium leaf. This is a pea tendril. This is really popular with a lot of the chefs. And then the edible flowers. Edible flowers are also really popular as well with the craft cocktail and the culinary scene. So that's another thing too. Edible flowers. That's been a huge. That's been a nice. If you get the edible flowers going, that's gonna be a nice complement to your product line with microgreens in the specialty in this specialized industry. This is an example of one restaurant. This is Mad. That sample of a pea tendril right there. And so when it comes to plating, it's a lot of the high-end restaurants, it's really about presentation. Let's see here. Let me try to get this here. Okay, this right here is an example of one of the craft cocktails. So this right here is an amethyst basil. We did not grow this to the microgreen size. So this would be an example where we have to reduce that seed density size to get the bigger leaf development, because they want that size, that structure to fit that craft cocktail just the right size. The micro size is just the first two cotyledons. This would be more at the petite/baby size. So the growth cycle is a little bit longer, but when we reduce the seed density, it's easier to get that development that the chef or the bartender wants. And that drink is really good, by the way. So if you're ever in Houston, check out Matt. I mean, it's really, really good. And the other thing that we do too that's a little bit different is microgreen art. So it kind of started out as the idea just kind of hit me when I was in the garage. I just wanted to do a restaurant's name in microgreens as a way of saying thank you for supporting us. And it was very rudimentary with how it looks, but when I took it to them, the response was really good. And I didn't even know what to call this stuff. I remember doing Google searches of doing microgreen art, mosaics with microgreens and I couldn't pull up anything. So I just started calling this stuff microgreen art. So now we do different full-blown mosaics and microgreens. We've done different corporate events. This is an example of the Houston Chronicle Culinary Stars that we did. And let's see here. That's another picture right there. There's another picture. So that's we got like some arugula, amaranth in there, some purple radish, some pea shoots. And that shouldn't go there. But and then so the biggest challenge on the microgreen art was figuring out how to time the growth cycles to get it to look like that, so everything pops at the time of presentation, because it is a living plant and they're going to continue to grow. So you have to stagger your growth cycles. That was one of the biggest challenges, getting that dialed in, and also the seed densities. When you go too dense, there's not enough air circulation, expect a lot of mold. And what ended up happening is I ended up showing my mom how to do this, and now she's just doing, I mean, her work is way better than mine. Even though I showed her all the techniques, now she's way better than I am at this stuff. So she's a pro when it comes to the microgreen art. So we've done, let's see if I can find, okay, let's see here. We've done, let's see here. Let me go back here. Micro Angelo is actually our microgreen art page. So that's when we do the events, and now it's asking me to sign in. So let's see here. I don't know why it's doing that. Let me see if I can pull up here. Nope. Didn't pull up. Well, it's micro.angelo. That's our microgreen art page. There we are. So Zest in the West. We've done some for other ones. We've got other ones that we've done. I just haven't uploaded them yet. But these are just some other examples. I took a lot of these pictures with my cell phone. So unfortunately, the quality isn't the greatest I like. Eventually, we'd like to get better quality photos. And you can kind of see the evolution where on the microgreen art, it doesn't look as clean or as crisp, but as the methods got dialed in, we figured out how to really refine these approaches and these methods on how to get things to pop at the day of different types of events. You know, when we, it's been a lot of fun to kind of see the evolution, the reaction. We go to these different events with the microgreen art, and that's something that's it's a fun thing to do. There's a lot of fun in what we do, but don't think that just microgreens and the biggest thing I can tell any new grower that's trying to get into this industry, while the barrier of entry is fairly inexpensive, it's a lot of work. It's not easy. You're really going to have to grind this out and really do a gut check on whether or not this is something you really want to do to scale this. Right now, we're a team of 7. We have mainly four of us that work inside the operation, 2 in accounting, part-time, and also Chef Connor, who's been doing a phenomenal job with the value-added items. We've got a lot of exciting projects in the works right now that we'll definitely be bringing to market in the very, very near future to really showcase how microgreens can change the game on a lot of things that we typically see in foods, and how their incorporation can be incorporated into different applications, from pestos, dressings, sauces, infused honeys. Our infused honeys are actually selling really well right now at farmers markets and other venues. And just different applications. We have one restaurant that's using some of our infused honey on their pizza. It's really interesting to see the response from the community, and just seeing these new ideas, and really staying in touch with the craft cocktail side, the chef community, the customer base. I mean, that's so critical. And just being a part of like a different type of niche industry like this, it has a lot of rewards, but again, I'll say this again, it's a lot of work. Every day, one of us is in the grow house doing something, stacking, unstacking, cleaning trays, the day-to-day grind, cleaning flood trays. We clean our flood trays once a month. So that entails having to disconnect everything, take it outside, power wash it down, because we have a lot of calcium. Even though we pull our water from a well and it goes through a multi-stage filtration system, it still has calcium. So the flood trays end up calcifying over a period of time. So what we end up doing is we'll actually have the flood trays. We'll actually have it up on a certain stand. We'll double stack them a certain way, and we'll actually have CLR sitting in a diluted ratio of CLR in the flood trays, and for about 8 to 10 hours. And then after that, then we scrub it to try to remove as much calcium as possible. Then we power wash it again. We use Xeritol 2.0 to sanitize the flood trays. So that way, each flood tray that's going back in there, it's clean, it's sanitized. Even the reservoirs, when we take those out and clean, and just the clean, the maintenance side of trying to scale an operation, that's a whole nother ball game unto itself. So while microgreens is very fun and exciting starting out, you got your wire rack. The daily grind on that stuff, it's something that cannot be overlooked. If you start falling behind on the daily grind on your cleaning, your maintenance, you're going to be inviting either different types of bacteria, pathogens, or just an unclean environment. And you really want to maintain a clean environment as much as possible when you're growing. And that's something that you're going to learn as far as what items are going to become higher probable items of what's going to get dirty a lot faster, and just maintain the consistency of those cleaning procedures. So,
Okay.
We really didn't have a blueprint to operate off of. We just kind of had to learn as we go. And we've really refined a lot of our standard operating procedures over time. We're going to be maxing out this grow operation in the very near future. So we're going to have to go into our next location. So a lot of the information here that from the previous speakers is very, very helpful for us. So, just stay tuned. I guess, how much time do we have?
Thank you. Thank you. This is great. Thank you. Can you hear me, James?
Yes.
Okay, yeah, please, you can stop sharing your screen. Thank you.
Okay.
I have a quick question for you. You mentioned NOP, which for those people who don't know that is the National Organic Program. Do you know what's the latest ruling on organic microgreens? Do you consider yourself organic?
Well, our practices is yes, and we actually have all of our record keeping in place. We were communicating with one of the representatives with NOP, and we told them as far as what it is we're doing, they don't really seem to have a format to work off of or a reference point for organically certifying an operation like ours. It's not to say it hasn't been done, but at least who we've been communicating with, it's kind of a new territory. So it's going to be, it seems to me that's going to be a learning curve for both, that when they do an audit and they check where we're sourcing our seeds from, how we're recording it, how the harvesting is from what's being sold as live, what's being sold as harvested.
Yeah. That's what I thought. But most importantly, are they allowing you to use media, like the potting mix media? Is that considered organic or what?
We do not. Our soils are OMRI certified. Yes.
Oh, okay. Excellent.
They are OMRI.
Excellent.
Yeah, they are OMRI.
All right. Excellent.
We've actually made some adjustments over the years, but all of our soils now are OMRI. A lot of our seeds are organic, but some are not, just because they are like none of the seed suppliers supply GMO seeds that we source from, but sometimes the availability of organic seeds is not there. And the other issue is that we have come across on at least in one particular case that the organic seed, for whatever reason, went to a sanitation type process, and the germination percentage was not as good as the conventional. So that's another consideration. While we try to source the organic as much as possible, sometimes the organic seeds just do not,
Yes.
like there was a wheatgrass one that we tried, just did not perform like the other ones. So we just couldn't, it was a loss of utilizing it, unfortunately.
Of course, as long as you record and why, and you try to make an effort to find organic
right.
seeds, things like that. Well,
Yeah.
again, thank you, James. Appreciate that, and good luck with your expansions and future work with ZP Organics. With that, let me share my screen. All right. This is the last presentation of this morning. We'll come back at 1. You can disconnect and reconnect at before 1 if you don't want to keep your computer on. I will keep my computer on. If you like to stay connected, that's no problem. But before you leave, I wanted to share with you. Do you see my screen with the Word file?
Yes, yes,
Okay.
we saw that.
With extension, evaluations is very important. That's how we rate how you like the program, how much you learned, what you would like to learn, things like that. You will receive an email. You will receive an email with this 5-question survey. So let me describe them quickly because sometimes they can be confusing. The first question is, did you learn? Did you increase your knowledge? Yes or no? Simple. The second question is you have to ask yourself, how much did you know before the meeting and after the meeting related to these 5 questions? So for example, hydroponic. If you knew nothing about hydroponic, then 1 is no knowledge or little knowledge. And let's say after the meeting, you say, yeah, okay, I learned more. I'm average now. Then you can select 3. So that's very simple. For these 5 choices here in question number 2, please select a number for your level of knowledge before the meeting and then corresponding after the meeting. Let's say you knew everything about supplemental lighting. You're an expert. So it's 5 before the meeting, and it stays 5 before the meeting because hopefully you did not unlearn something. Question number 3 is ask about what things you'd like to adopt and here you can select 1 or you can select all of them. Select anything that apply that you think about doing in the future. You're thinking about starting a hydroponic hobby system, you check here. Start growing microgreens, you check here. So select 1, select all. Question number 4 is asking you to give us an estimate of the economic value you assign to this information. We are not asking about how much you will be paying for future program, or if you select 100, then in the future we'll ask you to select $100. No, this is assign a dollar value to the value of the information you got. For example, let's say you want to learn how to make bread, and if you put a number value to your time, to the time you went to the store, to the time you spent on YouTube to learn how to make bread, and then you realize that loaf of bread is $20, then that will be like $20. It's cheaper to go buy it than to, for example, $20. So some people say, I learned a lot, I knew nothing. Boy, if I start a business, I can make $10,000 extra income, then you can select 10,000. Think along those lines, what is the value for the information? Another example I give is you get a plumber to come to your house, and that's $75 minimum just to come into your house. So something like that. Okay. And finally, question number 5 is on a scale of 0 to 10. 0 means not likely, and 10 means likely. Please select, would you recommend this to a friend, family, colleague? Would you attend the future program that we host in the future? If you like it very much and you have 0 issues, select 10 along those lines. All right. With that, I thank you again for being with us, and we will reconvene. At 1 o'clock.
Joe, there's some questions in the chat box. Do you want to answer?
Yes. Question number 3, my answer wouldn't fit the selections. That's fine. If none of them, of course, these are what we chose. This is not all the possible choices available in the world. So if none of them apply for you, just leave it blank. That's normal. That's fine. We will send you a link, so you will do the survey online. Everybody that joined, we have your email. Hopefully that when you registered, you enter your email, you will receive a link to this survey in the next few days.
What about the presentation?
The presentations are being recorded. Give us some time to edit and, fix it and clean it up and all that. And then we will send you a link of how you can download it or share let's say we put it on Google Docs, share the link that you can download it to view it in the future. Okay, I think this is all the questions related to the evaluation form. Again, thank you very much. Like I said, you're welcome to stay or disconnect and use the same link you connected this morning to join us again at 1 o'clock. Of course, please join us a little bit before 1 so everybody is admitted and you're ready to go. I have a presentation at 1 and then the 2 presentation by Eden Green, Mr. Aaron Fields. Thank you, and we'll talk to you later. Okay, so let's start with my presentation. Again, this is a collaborative work as a whole team. Dr. Niu, who presented this morning, is the lead in the urban horticulture program from the research with AgriLife Research. I'm with AgriLife Extension. And Dr. Hooks, who also presented this morning, is the research associate, and Ling Sun, the technician. We all work together on various research and trial and collecting data and this and that. All right, with background, some information. We know that growing leafy greens and hydroponics is gaining popularity. I hope you know that hydroponic saves water and fertilizer. The estimates are that you can grow the same crop of lettuce, for example, using 10% of the water in a hydroponic system as you would grown out in the field. And there is a potential for year-round production with hydroponics in a greenhouse. Of course, the greenhouse temperatures in southern states like Texas are too high in the summer for most leafy greens. Whether the red leaf lettuce is not red anymore, or it bolts too quick and it becomes bitter, or just plain doesn't germinate well. These are all issues with cool season vegetable when you try to grow them at hot temperatures. All right, and most greenhouse systems or greenhouse available now use the pad and fan or the water wall, if you're familiar with that term, as the typical cooling in the greenhouse. But this type of evaporative cooling is not very effective when the humidity is high, because the way this works is that it pushes the humidity from inside to the outside. And if outside it's hot and 100% humidity, the greenhouse and that water wall, the fan and pad cooling system is not very effective, which is typical in Texas, basically from June through September, is hot and humid conditions. So cooling the whole greenhouse air temperature is not possible unless you install air condition and incur the cost of an air condition. But cooling the root zone is an approach and can be energy efficient cooling method in these tropical and subtropical regions where you have hot and humid conditions in the in a greenhouse. Okay, so it is possible. And it's an idea that got us started thinking on those lines. And other researchers have done a little bit of work on that and have demonstrated that, for example, lettuce, the best root zone temperature is 75 when they were tested in the range between 54 and 88. Okay, so of course that the temperature effect is cultivar dependent. So if I tell you 75 is the best for lettuce, that does not mean it is the best for every single variety of lettuce. And of course, if it works for lettuce, It doesn't mean that it may be the same temperature for bok choy or for spinach or for any other crop. But even within lettuce, there are differences, as you will see in my presentation today. Okay, this figure here describes again the effect of temperature on growth rate and what happens within the cell at the cellular level. Okay, we all know that life slows down. Even at the molecular level, enzymatic reaction, even the molecule starts moving slowly with colder temperatures. So there is a minimum where life will completely stop, or even death can occur if the temperature gets real low. Okay. So that's the minimum. I guess some people say it's the gel, the formation in the membrane or whatever. But as the temperature goes up, the enzymes and everything that goes with it responds linearly. As the temperature goes up, the growth rate responds linearly until you reach an optimal or the maximum rate, where we say that the enzymatic reactions are working at the maximum potential. If you continue to raise the temperature, you're not going to get any more benefit, and it's not going to even slow down or plateau. Actually, if you continue raising the temperature above that optimal, you can have a negative effect. The proteins start to break down, the cytoplasm or the cell membrane starts to break apart, and you can even reach death. I mean, it may not, this is a schematic and this is symbolic. It doesn't, it may not be that drastic, like within 5 degrees, it goes from optimum to death. This is just a descriptive figure. Okay, this is a realistic display of basal response to air temperature. Work done in 2016 by Walters and Curry from Iowa State University. You'll see that at air temperature of 52, the basal it's just sat there. I bet you this is like 3 weeks old, just sat there, because for this one to reach this height, that's 2 or 3 weeks at least. So that just sat there. It didn't kill it. It's not dead. It can, but it's barely moving. 63 a little bit better, 78, 84. You see that linear response. Now, like I said, if you go above the maximum, it's not gonna instantly kill it. So 95, it's slower. I bet you maybe, I'm guessing maybe 100, even a little slower. Who knows? Maybe 120 will completely kill it. I'm guessing. But that shows you the ideal temperature, air temperature, and response to growth, to plant growth, to increasing air temperature. And similar thing can be observed with the soil temperature or with the water temperature when we are talking about a hydroponic system. So the objective of this study, we wanted to determine if the reservoir temperature, the temperature of the reservoir nutrient solution, can influence the growth of various leafy greens in a deep water culture system in a greenhouse. And our hypothesis that we started with this work is that, yes, cooling the solution temperature will improve the plant growth. So this is our hypothesis that it will work, and we want the data to disprove us or prove our work. Okay, the setup was very simple. We built a 4x4 metal frame and we insulated it with foam and with a liner, and you'll see the black clips to hold the liner in, and we filled it with the fertilizer solution. So that's about, I guess, 10 inches deep. And 4x4, and we bought the foam boards with the holes pre-made in them. And this here is the Active Aqua water chiller cooling system. Here's a description of it. It's a 1/4 horsepower rated at 3010 BTU per hour. And one pipe with the water in, and another tube with the water goes in, gets chilled, and gets back in the system. My estimate for 4x4 by about 1 foot depth of water is that each one of these holds about 120 gallons of water. And of course, you program it and you set it at the temperature desired. And here we set it at 75 degrees Fahrenheit for the water temperature. Let me take a tangent and talk about, in case you are interested in this and you want to buy a water chiller, you have an idea of what size unit you need to buy. For that, you need to know your temperature differential, like what is the actual temperature and where you want it to be. So what's the difference in that temperature, and the flow rate of that chiller, of the equipment you're buying. So if the temperature differential is equal to the incoming temperature, the ambient temperature, minus the desired, what's that desired temperature you wanted? So you multiply that differential by gallons per hour, which is the flow rate, times 8.33, which is pounds per gallon of water. Each gallon weighs 8.33 pounds, and that gives you the BTU per hour. You divide that number by 12,000, and that gives you the tons of cooling capacity. Many businesses understand that number. If you tell them, I want a chiller or a cooling or a refrigerator or something with 1 ton or 2 ton or half a ton, they understand that system. Of course, many also can understand the BTU per hour. To be safe, add about 20% for your ideal size of the system you wanted to buy. So based on that, the chiller, the Aqua that I showed you, the one we were using, here's the information that we got from the manual. The flow rate of it is between 132 to 396 gallon per hour. I'm choosing 132 in my calculation. And it is rated at 1,040 BTU per hour. Divide that by 12,000, that's 0.09 tons. Okay, let's play with some numbers. The water temperature was 86. You'll see that later in experiment 1. The desired temperature was 75. Actually, it was 74. So let's work with the real numbers. 86 minus 74. So the temperature differential, I'm dropping the water 12 degrees. So what's the BTU? 132 gallon per hour times 8.33 times 12 degree differential. That's 13,195 BTUs. Divide that by 12,000, 1.1 tons. You add 20%, that's 1.3 ton. If we wanted something that can chill the system in 1 hour, we needed to buy a unit that is rated as at 1.3 or 1.1 ton. The one that we bought is 0.09. So it would take 12 hours if you divide 1.1 to 0.09 initially from the time you set it up. It will take about 12 hours to reach that desired 12 Fahrenheit differential and drop the temperature from 86 to 74. And then after that, it will continue running to, of course, it may not run continuously, it will on and off to keep that temperature at the desired level. So this is a tangent in case you're interested in this topic, and especially how to calculate what the tonnage of your cooling system capacity. All right, so back to our greenhouse and to our system. You see here's the greenhouse with the pad and fan, the pad on one side, the fan on the other end, what I call water wall. A lot of people call it water wall. 4x4 each of these units. We replicated it 3 times. For science, you need replication, you need to repeat. You cannot just do one sample and do statistics with it. So on the left side are the 3 replications for the chilling. And you see we had the foam insulation on the water pipes going in and going out to reduce water loss. I mean, to reduce temperature loss. And on the right side are the control treatment. Unchilled control, the water temperature is the ambient, whatever the water temperature is for that time of the year. Okay. Again, as a side note, it seems to me that whenever I plant any kind of cool season crop, worms comes with it, as if the worms' eggs are on the seed itself. Be prepared, whether in the garden, whether in a hydroponic, aquaponic, any type of system. If you plant any, make sure you have BT ready, and at the sign of the very first hole, start spraying. Luckily, the worms are small and they don't move, and they kind of focus their efforts on one plant. So when you see something like this, look everywhere, inside, outside, until you find it and you get rid of it. Okay, or you can spray if you don't scout every day and you miss and you see more than one plant present, and then time to spray. With water spinach, which is a crop we tested only once, but this is such an interesting result. I wanted to share that with you. If you see this is not fungus, this is not mold, which what I thought it was first. Actually, this is the minerals, the nutrient that came, and they were so concentrated and accumulated and busted the cells and oozed out. And as they dried out, these minerals from the nutrient solution crystallized on the surface. So to me, then water spinach, which is what this is, water spinach, then needs a lot less fertilizer to use and to avoid such an issue. Okay, anyway, just for information. All right, let's get into the three little experiments we did this year. The first one between July 8th and August 5th. Again, that the chilled temperature was set at 75, and the unchilled or control was the ambient water temperature. We started with 7 crops. Water spinach was one of them. I just showed you one leaf of that. 2 varieties of spinach, seaside and mandolin. 2 varieties of pak choi, white stem and purple magic, and 2 varieties of lettuce, red mist and Bergen's green. The daily average temperatures are shown here. The greenhouse air is 82. The ambient water temperature, the control was 86, and the chill treatment, the water temperature was 74, and that's the average, of course, even though our setting was set at 75. So, and those two are the numbers I just showed you in my calculations, 86 and 74. So there is 12 degree differentials. That's definitely big enough. If there were to be any effect of the temperature of the water, it should be here. Don't expect to see differences if the differential was, let's say, 4 or 5 degrees. I doubt you'll see any effect on that. So again, from left to right, the names are listed here at the bottom. We had 8 plants of each crop, each variety. So you have the water spinach, followed by the seaside spinach and mandolin, then the white stem and purple magic pak choi, and then the 2 lettuce, red mist and Bergen's green. This picture was taken 7 days after initiation of the experiment, 7 days after treatment. And the only thing that jumps out and you can clearly see is in those red rectangles where the 2 spinach performed a lot better in the chilled treatment compared to the control treatment where the water temperature was about 86 on average. We know spinach is a cool season crop. It does not like hot temperatures, air temperature. It grows better in the winter garden in Texas where you have cool and mild winters, and that's evident here. That it prefers cooler water even though the air temperature was warm enough. 14 days after treatment, of course, in addition to the spinach, where the spinach, the two varieties in the chill treatment continued to perform, and some of them completely died when in the control treatment. That's still clearly obvious. But what's interesting is that the water spinach, which is a tropical heat-loving plant, I mean, I've seen it in Texas, south of Houston, growing in the summer inside the greenhouse, and it was doing great. So it loves the heat, and it's evident here that the water spinach preferred warmer water temperatures than cooler water temperatures. Everything else visually, it's not very clear. We had to get weights to be sure one way or another. When you lift up the foam boards and you look at the roots, in general, we found that most of the crops, except on the top, which is the water spinach, preferred the cooler temperature. You see the intensity of and the how many more roots are in all those crops compared to the control with the warmer water. The water solution is pinkish. That's the nutrient solution. And by the way, we did not add air to that. To the water solution. So let's look at some data here from experiment 1. We are looking here at bolting. Basically, we measured the length of the stem, and the longer it is, it means it bolted. You'll see that for lettuce, Bergen's green bolted, and the stems were longer than red mist. So different species of lettuce responded differently to the temperature. And there were no difference between the control and the chill treatment. So the pink is the control, little higher, little longer, but not statistically different. If you don't see different letters like you would see here for Purple Magic, then these differences are not statistically important. They're just random differences due to chance. And so that's one observation, is that lettuce, different varieties of lettuce responded differently, but no effect on the chilling on bolting. So in other words, Bergen's green bolted more, tends to bolt more than red mist, and chilling the water did not fix that problem. It still bolted more. With pak choy, again, we saw the same observation that the Purple Magic responded differently than white stem. But here we did observe where chilling the water for Purple Magic pak choy did reduce the bolting significantly than the unchilled water temperature. So it seems here there is benefit to chilling the water if you are growing bok choy. And with lettuce, I would tell you switch to a different variety that's naturally resistant to bolting, because it doesn't seem like chilling the water had any effect on it. Mandolin and Seaside, I told you they died, or most of them were died, so we didn't take data for here. We did take fresh weight of the shoot fresh weight. And here, again, the chilled, even though there was no statistical difference in the bolting, the Bergen's green had a little higher, but statistically different, higher fresh weight when the water was chilled. No statistical differences in the red mist fresh weight between heated, ambient and chilled water temperatures. Purple Magic, no differences for Purple Magic in terms of shoot weight, but again, when we chilled the water, we had more harvestable yield or shoot fresh weight for white stem. Okay, and of course, since the spinach died or almost was completely dead with the controlled temperatures, then they, even though the chilled one looked miserable and definitely not marketable and poor quality, but there was still enough weight to give you statistical differences. All right, experiment 2. Is conducted August 13 to September 2nd, following the first one. And here are the temperature dropped a little bit. Instead of 82, 86, and 74, the greenhouse temperature was now 80 on average, and the control tanks had 84 instead of 86, and 73 instead of 74. Okay. And this is right before harvest. Here you see that water spinach is not present anymore. We still use the same 6 treatment, the same 6 other crops, 2 each of spinach, bok choy, and lettuce. But this time they're randomized here. So at days 13, you still see the difference, especially in the spinach, seaside and mandolin, where they're bigger, and then in the warm water temperatures. Other stuff, yes, if you look closely, but you can guess one way or another. We had to take actual data to look, and I'll show you that. Again, here we saw that with the chilling the water, reducing the water temperature, we had better growth in the roots than in the warm water temperatures. So let's look at the shoot fresh weight. Here again, you'll see that there are similar trends where the what do you call it? Burgund's green, the chilled, had better yield than the control, and the Burgund's green had, in general, better yield than the red mist. Okay, bok choy. So these are similar results as experiment 1 in the sense, in generalities. The only difference here is that in mandolin, here, I'm sorry, Purple Magic had better yield in the control, but in experiment 1, these were the same, not statistically different. Okay, but the same generalities with experiment 1 is that there are species or varietal differences where white stem did better than Purple Magic. And of course, mandolin and seaside, without a question, chilling the temperature, chilling the water benefits, but still not to the level that reached the marketable stage. By the time we reached study number 3, it was cool. So we decided we weren't sure if we will get really the effect of warm season on the air and the water temperature. So we decided to work with just 6 varieties of bok choy. And here are the average daily temperatures. So now the greenhouse air temperature is 74 instead of being in the 80s. And the water temperature of the control is 78 instead of, again, in the mid to upper 80s. And now the water temperature of the chilled solution is 68. So again, we wanted to have at least 10 degree difference to see any effect. We could not keep it at 75. Otherwise, like I said, 75 to 78, that's not going to have any effect. So we dropped the water temperature, hopefully to see some, to see if we can get any good effect. And here are the 6 varieties of bok choy listed: Ching Chong, Petite Star, Purple Magic, Tatsoi, white stem, and white stem tatsoi. And again, the same setup, 3 chilled tanks for replication for statistical analysis, and 3 control tanks unchilled, just whatever the water temperature is. And here how they looked at day 15 at harvest, about 2 weeks later. So this is proof I could have shown you for for the first 2 experiments. This is just a proof that the measurement of the water reservoir temperature was significantly different between the 2. They were set and they are what they are because we were taking the measurements, you know. So of course there's plus or minus 1 degree between one tank and the other, but on average, no overlap and big difference between the 2. And air temperature over all the 6 tanks varied a little bit, plus or minus 2 degrees. So that's considered acceptable variation. pH and EC. For this experiment, we observed that the control in red, the EC, was consistently higher than the EC electrical conductivity of the chilled solution. The pH was similar till about 10 days, when the chilled solution started to have lower and lower pH from about 6.2 to 5.6. You know, half a point difference. If this was aquaponic, I'll tell you, hey, the plants are doing better. Lower pH, you know, nitrification is happening, something more, but this is hydroponic, and I'm not an expert to explain why the pH dropped here. All right, this is how the 6 pak choi varieties looked 7 days after initiation of the trial. On the left here, you see the chilled treatment, and on the right, the control. Not really anything that can jump right at you and tell you this is better than the other. Maybe here, the tatsoi, a few of them are bigger than a few of them here on the left, but that's not what we want to go by, because 2 weeks later, here you may, if you look closely and you stare long enough and you look at, you may be able to see some differences, like the white stem, definitely thicker and bigger leaves in the control and in the chilled, and you know. But again, the final answer and the accurate answer is you have to take weights instead of visual cues. Again, no obvious differences this time of year between the chilled or the control treatment. Unlike the first 2 experiments where I showed you that chilling the water had better, had more and bigger roots, even though there was no equally corresponding bigger shoot fresh weight. But here at this stage, with these 6 bok choy varieties, there are really no obvious differences. But we did take root weights and measured them, and I'll show you the data here. So the top part is the shoot fresh weight. And again, when you see 2 different letters, means these are statistically different. The red is the control, the blue is the chilled. So for all 6 bok choy varieties, the plants growing in the control treatment unchilled were bigger and resulted in more fresh weight than in the control treatment across all 6 varieties. And remember that the chill treatment was 68 Fahrenheit on average, and the control water treatment was about 78 degrees Fahrenheit. For the roots, on the bottom part, for the roots, only Ching Chang and white stem, these 2 here, showed statistical difference. The other 4, you know, plus or minus, but nothing statistically obvious. So we concluded from this experiment number 3 is that 20 degrees Celsius or 68 Fahrenheit is too low for optimal root development and plant growth of all 6 bok choy varieties that we tested, compared to 25 degree C that's in the control. The overall conclusions that the chilling, the hydroponic solution has potential for cool season leafy greens, as evidenced in the spinach cultivar and the results that we observed. And then more variety trials are needed since temperature response is both species and cultivar dependent.